Welcome, everyone. Welcome to our biology um, session, 3-hour revision session where we're gonna go over as many topics as we can, inshallah, to help you guys prepare for your exams. So, this is our Academy X. I know most of you attended our session yesterday as well, so you guys are already familiar with it. So our Academy X has been teaching IGCSE, A levels, IB for about over 15 years now. I'm going to be teaching you guys today. I am Dr. Fatma. I have been teaching IGCSE and A levels for over 10 years plus now. And I really hope you guys are going to go ahead and you guys are going to really enjoy your session today. We are starting, by the way, our next year's batches. We're taking admissions for the next 2025 May June exams. All our students, they get one-on-one classes, or we take a maximum of 3 students in a batch. We do not take more than 3 students, which is why we have very limited seats. So we take on one-on-one classes, or you can take a batch classes. We prepare you topic by topic. Each topic, we follow it up with past paper practice. And please go ahead and do take a screenshot of this. This has the website. This is the website where all the resources are uploaded. So over here on this website, you're going to be given access to all the resources. Towards the end of the session, we will give you the access. So stay tuned for that. Take a screenshot of it. It also has the number in case you have any queries or anything that you want to go ahead and ask. So today we are taking a look at our biology syllabus. And as you guys know, biology syllabus tends to be quite extensive, right? I think from physics, chemistry, biology is the one that's the most extensive and the hardest to cover because it's the longest. So we are going to go ahead. We're first going to start by taking a look at all the topics related to genetics, because I feel like those are the topics that can be a bit confusing. We'll start by taking a look at inheritance. We'll go over our variation and selection. And we're going to take a look at biotechnology, genetic engineering, which are some actually pretty interesting topics as well. And then later on, we're going to go ahead and take a look at other topics as well, like you have got transport in animals, you've got your diseases and immunity, you've got the topic of excretion, coordination, and response. So we'll go ahead and we'll take a look at that as well. Again, the topic is syllabus for biology is quite long. I really wish I could go over the entire syllabus, but what I've done is I've chosen some really important topics and we're going to go over them. Okay. Now,
Miss, can you also give us
yeah.
the key information about past paper and solving them?
Yes, I will. We will. We will go over how to solve some questions as well, definitely.
Thank
All
you.
right. Now, so when we are let's first of all start with the first part of our topic, which is obviously going to be the topic of inheritance. I've started with inheritance first because it will lay down the basics for the entire topic of your genetics. The entire genetics will make sense if you understand how inheritance happens. Now, for us to understand inheritance, right, we first need to understand a little bit about DNA. You guys might have heard so many times the terminology DNA. You guys might have heard so many times chromosomes. Now, what does this all actually mean? That is the important thing. To understand this, think of it in this way. DNA is a molecule which has got a double helical structure. So a DNA has got 2 strands. All right. These strands are made up of 3 components. Okay. I'm telling you the monomer. I'm telling you the Lego piece, right? The Lego piece. The Lego piece is that you have a sugar. Okay. To this sugar, we have a phosphate group. And in front of the phosphate group, we have a nitrogenous base. This is the unit. This is the basic unit, the Lego unit, right? I will join multiple Lego units together to give me the entire structure of DNA. So this is the Lego unit. We call it a nucleotide. What is the name of it? We call it a nucleotide. Now, it's a Lego piece. What is going to happen that multiple nucleotide will join together with each other. Multiple nucleotide will join together with each other to make a strand of DNA. Okay? Just like that, we get one strand and then we get another strand. So we will end up getting two strands of DNA, right? DNA includes two strands. What are the three components of it that I want you guys to remember? The three components are, we have a sugar component. The sugar is actually deoxyribose. The sugar is known as deoxyribose. What does deoxy mean? Deoxy means it has one less sugar. One less oxygen. What does ribose mean? Ribose, you guys must have gone over the topic of biological molecules. Ribose is a monosaccharide. It's a sugar. It's a carbohydrate. It's a monosaccharide that has 5 carbon atoms inside it. So what is deoxyribose? It's a monosaccharide. And it's a monosaccharide that has only 5 carbon atoms. Now, so the sugar will be deoxyribose. Along with that, you are going to have a phosphate group. And along with that, the third component you're going to have is a nitrogenous base. Now, this is the monomer, okay? The monomer, all three of these together lead to the formation of a monomer, which is a nucleotide. Now, multiple nucleotide will make a bond with each other to give you your structure of DNA. Multiple nucleotide will come together to give you your structure of DNA. Okay, if everything is clear so far, let's talk a bit more specific. When we talk about our nitrogenous base, right? So in our nitrogenous base, there are 4 types of bases that we can have. We can have adenine. We can have guanine. We can have cytosine. And we can have thymine. Now, the thing about these, so there are 4 possible nitrogenous base. So this nitrogenous base can be any of these 4. Any of these 4. Now, what is going to happen is this. That these bases, this is DNA, okay? DNA is a double-stranded structure. What does a double-stranded structure mean? A double-stranded structure means that you have got 2 strands of DNA that are facing each other, right? Now, what happens that these bases, these nitrogenous bases, adenine, guanine, cytosine, thymine, these nitrogenous bases, they tend to pair up with each other, right? What does that mean? That means that adenine is always going to be 1, 2 in front of thymine. Adenine will say, listen, wherever there's thymine, I want to be in front of it, right? Adenine and thymine go hand in hand. They're like the best buds. If we have adenine, so which nucleotide is going to be in front of it? Thymine. And then cytosine and guanine. Cytosine and guanine go hand in hand. Wherever we have cytosine in front of it, we will have guanine. So we have adenine and thymine and cytosine and guanine. This We have a term to explain this. We have a term to explain this friendship. We have a term to explain why these two are always in front of each other. We, in the biology, when you guys are answering questions, you're going to say complementary base pairs. These two are complementary to each other. They like to be next to each other. These are your complementary base pairs. Right? These are your complementary base pairs that actually like to be in front of each other. So in front of adenine, we have thymine. In front of cytosine, we have guanine. And that is the exact trend you guys are going to see throughout the entire structure. Then, when we talk about our adenine and we talk about our thymine and even cytosine and guanine, right? When we go ahead and we are going to talk about it. Remember, the bond that forms between these base pairs, between the nitrogenous base pairs, this is a hydrogen bond. Okay? These nitrogenous base pairs, they all develop hydrogen bonds. These nitrogenous base pairs, so when they're going to arrange themselves on top of each other, when they're going to arrange themselves in front of each other, they're going to develop hydrogen bond. Okay? So this is what the structure of DNA is going to look like. At the on either side, you will have a backbone. Okay? On either side, you will have your backbone. This is the backbone of the DNA. The backbone of the DNA is made up of this part of the nucleotide, the sugar and the phosphate. So the sugar and the phosphate will make the spine of the DNA, the backbone of the DNA. In fact, that's exactly what we call it, right? We actually go ahead and we call it sugar phosphate backbone. So a DNA is going to have a sugar phosphate backbone where the ribose sugar, deoxyribose sugar, and the phosphate group are going to make a bond with each other, and you will get a long chain. In front of them, you're going to have your nitrogenous bases. Those nitrogenous bases can either be adenine or guanine or cytosine or thymine, depends upon the person's genetic information. The nitrogenous bases are going to develop hydrogen bond based on the complementary base pairing. So if you have adenine in front of thymine, if you have adenine in front of thymine, so they will be a base pair. And cytosine will make a pair with guanine, and you will have hydrogen bonding between them. And then finally, what's going to happen that these two strands, once they're all ready, they will wrap around each other and they will give you a double helical structure. This is how the DNA is actually made. This is how the DNA is formed. We start from a nucleotide, the nucleotide join together, they give you the DNA strand. The two DNA strands are going to be arranged in such a way that they will be complementary to each other. The strands, the nitrogenous base pairs will have hydrogen bonding. And as a result, you're going to go ahead and see that the two strands will arrange next to each other. They will wind around each other, lap around each other, and give you a double helical structure. So this is how DNA is actually formed. Now, DNA, structure of DNA is something that you might be familiar with, right? But when we talk about the structure of a DNA, when you talk about inheritance, because we are right now talking about inheritance, right? When we talk about inheritance, you must have repeatedly heard the terminology chromosome, 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 chromosome. What actually is chromosome? Let's discuss it. Now, when we talk about the DNA, with the DNA, there's a small problem. The problem is that a single strand of DNA is so long that you cannot fit it inside a small, teeny tiny nucleus, right? The nucleus of our cells is really small. You cannot see it within the microscope. So a DNA has so much genetic information on it. It's really long. So how does it fit inside the nucleus? Now, To understand that, we have to realize that the DNA is not found lying openly within our nucleus. It's not just lying there. No. What happens is this, that the DNA, right? Let's draw this 1 second. What is going to happen is this, that my DNA, my double-stranded DNA, is actually going to start wrapping itself around some proteins. Okay? DNA is not found loosely lying around. No. What DNA does is that the DNA starts to wrap itself around some proteins. Okay? Like this. These proteins are known as histones. So the DNA is going to wrap itself around histones. And what is the result of this? What's the point of the wrapping? The point of the wrapping is basically that all the DNA is going to coil and come together and wrap around each other, and it will end up making a thread-like structure. That thread-like structure is known as chromatin. Chromatin. This is what is found normally within the nucleus. If you guys take a cell, put it under light microscope, this is what you guys are probably going to end up seeing. You guys are going to see this is your nucleus. And inside the nucleus, all of the DNA is lying like this, like a loose thread, right? So DNA itself is quite long. DNA itself is quite long. So it will wrap itself around the histones. It's going to coil, it's going to tangle up, not tangle, but you get the idea. It's going to wrap, it's going to go around, and it's going to give you a thread-like structure, which is known as chromatin. So chromatin is the same thing as DNA, but now that DNA has, sort of wrapped itself together. If you take a piece of thread and you tangle it all up, it becomes smaller, right? Something along those lines. Now, this is how you will normally find DNA within the cell. You'll find DNA in the form of a chromatin. But when the cell is about to divide, this chromatin will start to coil up. This chromatin will start to coil up. It will start to wrap around itself. It will start to, come together. And all of the chromatin will condense and give you your chromosomes. So chromosomes are simply the condensed form of DNA. All the DNA has been condensed, it has been put through, it has all been condensed, and it's all come together to give you your chromosome. That is what chromosomes actually are. That's how they're formed. And point to be noticed, chromosomes are only formed when the cell is dividing. If your cell is not dividing, you will not see any chromosomes. In that case, you will only be able to observe chromatin. Okay? So chromosomes are only visible when the cell is about to divide. So this is what a chromosome actually is. Now that we know what a chromosome actually is, let's talk about the different parts of chromosomes. Okay? So when we talk about the different part of a chromosome generally has two arms. Okay? So, okay, now what is going to happen that in our chromosome, there are two parts of a chromosome. Okay? This is one part, and this is the other. The half of a DNA, one half of a chromosome, this one half of a chromosome is known as chromatid. Okay, so this one half of a chromosome is what we call as our chromatid. Okay, both of these chromatids, we call them sister chromatids. If you guys go over your textbook, you might often say that they say sister chromatids. Why do we call them sister chromatids? Because they're identical to each other. Each chromatid has one copy of the DNA. So one copy of the DNA is in one chromatid and the other copy is on the other chromatid. So when the cell is going to divide, one chromatid will go to either cell. This chromatid will go to cell number 1. This chromatid will go to cell number 2. So this way, both daughter cells will get one copy of the DNA. So this chromatid has one copy, this chromatid has one copy. Okay? And when they're going to break away, a copy of the DNA will pass to both the cells. Now, the total number of chromosomes are
Can
very
you repeat the last part?
Yes, I will repeat the last part. Also, we will discuss mitosis, the steps of mitosis. So it will make a bit more sense then.
Miss?
When you talk about, yeah, is someone saying something?
Yeah, Miss, sorry to interrupt, but will you go over protein synthesis as well?
Yes, Aram, I'll try my best to go over it.
Okay, Miss, thank you.
Okay, so now what is going to happen that each chromatid has got one copy of the DNA. Okay, so what is going to happen that when the cell is going to divide, this chromosome breaks in two. I'll go over the steps of mitosis, so we will discuss this in more detail. But when the cell divides, right, the chromatid or the chromosome breaks into two portions like this, right? So you have one chromatid over here and one chromatid over here. Okay. Now, what is going to happen that since each chromatid has a copy of the DNA, right? Both chromatids have one copy of the DNA. So when the cell is going to divide, right, the chromatids will separate away. One chromatid will go to one of the daughter cells. This will go to one of the daughter cells, and this will go to another daughter cell. So this way, both of the daughter cells will get one copy of the DNA. Okay, so both these are known as chromatids. Both have one copy of the DNA. So each will have one
mitosis,
copy. Sorry?
It's mitosis.
Yes, exactly. This is mitosis. Yes, we're talking about mitosis. So this is exactly what a chromosome is. This is what a chromosome is. A condensed form of a DNA. And it's made up of two chromatids, and both chromatids are going to have one copy of DNA inside them. Now, when we are going to go ahead and we are going to talk about it, you guys should know that the number of chromosomes in every species is very important. Every species has a specific number of chromosomes. Okay? If that number of chromosomes, a specific number of chromosomes, and that can actually help in identifying what a species is. For example, humans have a total of 46 chromosomes, 46 chromosome or 23 pairs. Okay, 23 pairs or 46 chromosomes. So in total, if you start counting 1, 2, 3, 4, 46. But if you talk about pairs, so there are 23 pairs. Okay, when we talk about chromosomes, chromosomes are found in pairs. By the way, just so you guys know, chromosomes are always found in pairs. And we will talk about later on why that is. So when we talk about chromosomes, our chromosomes can be divided into two categories, right? We can have the chromosomes that are known as our sex chromosomes or our gender chromosomes, right? The sex chromosomes X and Y are the ones that are actually going to be deciding the gender of the offspring. So we can have our gender chromosomes or sex chromosomes, and we can have the autosomal chromosomes. So everything other than the X and Y chromosome, anything else other than X and Y chromosome is going to be our autosomal chromosome. Okay, so now we know what chromosomes are. Now, how are chromosomes related to inheritance? Before we go over the genetic portion of it, let's talk about the cell division portion of it. Okay, when we talk about the division of our cells, so cells divide by mitosis, right? Mitosis, you guys will see questions in your past papers where they will ask you why mitosis is so important. So what are the points that you guys can list in questions like that? You are going to list that mitosis is important because mitosis helps in growth. If I want to make new cells, if I want a tissue to grow, I need mitosis to occur. Then we can, because for repair of a tissue, if a tissue has undergone any damage, if it's not, if there's been any injury to it, for the repair of a tissue, I need the cells to undergo mitosis. Then, when we're going to go ahead and talk about it, mitosis is very important in, one second. It helps in formation of new tissue. It helps in increasing the number of cells. Right? So mitosis, wherever I need new cells, wherever I need there to be any kind of repair, any kind of growth, any kind of division, I need mitosis to occur. Now, when we talk about mitosis, what are you guys expected to know in your exam? You guys are expected to know exactly what mitosis is and what are the steps related to it. Increasing the number of cells. What are exactly the points related to it, right? So first of all, what is mitosis? Mitosis is the division of the cells to give you two daughter cells. Okay, so this is division of the cell in which a cell is dividing to give you two daughter cells. In mitosis, the DNA remains the same. The amount of DNA remains the same. I will explain what I mean by that. But before I explain what I mean by that, let me introduce you guys to two terminologies, haploid and diploid. Now, what is a diploid nucleus? A diploid nucleus is a nucleus that contains two sets of chromosomes. Okay? A diploid nucleus is the one that contains two sets of chromosomes. So that is known as a diploid nucleus. Okay? So for example, how many chromosomes do we have in our body inside the humans? We have 46, right? Then we have a haploid. What is a haploid nucleus? Haploid nucleus has got half the number of chromosomes. So if the total number of 46, the number of chromosomes are inside the number of chromosomes inside the haploid nucleus are going to be half of that. So they are going to be 23, right? Now, what happens is this, that the reason I'm emphasizing this point is because we also need to go over meiosis, okay? In meiosis, the number of chromosomes changes as the cell divides. But in mitosis, the number of chromosomes remain the same. So if the number of chromosomes in my body are 46, then when the cell is done dividing, the daughter cells will still have 46 chromosomes. Okay? And that's a really important point. So in the mitosis, the number of chromosomes are constant. They do not change. Now, when we talk about mitosis, we have to talk about the 4 stages of mitosis. The 4 stages of mitosis can be broken down as prophase, metaphase, anaphase, and telophase. What is prophase? So, do you guys remember how I told you that all of the DNA is lying loosely in the form of some threads that we call chromatin? Right? That's what I told you guys. All of the DNA is lying loosely in the form of your chromatin. So, What is going to happen before the cell can divide? That chromatin has to condense and turn into chromosome. And that's what you will observe in prophase. In the prophase, you guys are going to see that the chromosomes are going to appear. Your chromosomes are going to start to appear in your prophase. Then, all right, so the chromosomes will be visible. Now, when you guys will stay in the cell, you'll see such nice chromosomes, right? You might even be able to see them slowly forming. So you can see the chromosomes forming. Then another thing that's going to happen is this, that your nuclear envelope is going to start to break down and it will start to disappear, right? Your nuclear envelope is going to break down. Your nuclear envelope is going to start to slowly disappear. You will not be able to see the nuclear envelope, the clear membrane around the nucleus anymore. Then the third thing that's going to happen is you have your mitotic spindle, right? Mitotic. So mitotic spindle has to. That mitotic spindle forms because of your centrioles. Centrioles are small organelles. They're small structures inside the cell that form the mitotic spindle. I will explain to you guys what the mitotic spindle is. But centrioles are the ones that are necessary for that spindle to form. So the centrioles will go to the opposite side of the cell. The centrioles will move towards the opposite side of the cell. All three things are going to be visible for you guys in prophase. So see, this is exactly, you can see what's happening in prophase, that just before the cell is going to divide, chromosomes are appearing. See, condensation of the chromosomes, right? And then what is going to happen? That the nuclear membrane is going to disappear. The nuclear membrane is going to disappear and centrioles are going to go to the opposite corners of the cell. Now, what happens after that? What happens after that is the next stage is known as metaphase. What happens in metaphase? What happens in metaphase is this is my cell. Now, in metaphase, remember how I told you guys about the centrioles? So the centrioles will produce a thread-like structure. It's going to look like a thread-like structure. Okay? They're going to produce a thread-like structure that we call the mitotic spindle. This thread-like structure is known as the mitotic spindle. On the mitotic spindle, in the very center of the cell, right where the center of the cell is, all the chromosomes will arrange themselves in one line. All the chromosomes will arrange themselves in one line like this, right? All see, they're in one line and right towards the center of the cell. This is metaphase. So in metaphase, the mitotic spindle is going to form. That's the first observation you guys are going to see, that your mitotic spindle is the thread-like structure that helps in mitosis. So mitotic spindle will form and all the chromosomes are going to line up. They're going to line up right where the center of the cell is. This is metaphase. Now, what happens in anaphase? In anaphase, now what is going to happen? That these thread-like structures will start pulling the chromosomes apart. That's what they're going to do. The thread-like structures will now start contracting and they will start pulling these chromosomes away from each other. So remember a chromosome, what was a chromosome? A chromosome was made up of two chromatids like this, right? This is one portion of it and this is the other portion of it. It can break in the center. And when it breaks in the center, it gives you two chromatids like this. So just like that, the chromosome is going to split in the center. And when the chromosome is going to split in the center, half of the chromatid is going to go to one side and half of the chromatids will go to the other side. And this is because the mitotic spindle is contracting. The mitotic spindle is contracting, it's pulling, right? So, and that's what you're going to observe. You are going to observe that the spindle will pull apart the spindle will pull apart the chromatids. And half of the chromatids will go to one side of the cell and half of them will go to the other side of the cell. The final step, now what has happened is that half of the chromatids are on one side of the cell, half of them are on the other side of the cell. So now the DNA is equally divided between the two cells, right? Now, the final stage that we have is telophase. In telophase, now that the DNA is equally divided, the cell is going to split into two. Half of the chromosomes or half of the chromatid is on one side and half of it is on the other side. Now the cell is going to squeeze the center. It literally squeezes. That's exactly how it happens. The cell squeezes from the middle and it separates it out into two daughter cells. And once the mitosis is complete, the chromosomes will again disappear, they will loosen up back into chromatin, and the cells will start doing the normal function that they normally do. Mitotic spindle, Tia. Spindle. I think that's what you were asking. Spindle is S-P-I-N-D-L-E. So this is what happens in mitosis. So mitosis is the cell division that that happens everywhere in our body for growth, for repair, for replacement of cells, right? Even when it comes to asexual reproduction, which is what we see, for example, with bacteria. Bacteria divides asexually. That's mitosis. You have got plants that divide asexually. That's going to be mitosis as well. So mitosis has a lot of different properties. Then, along with mitosis, we have another type of cell division, and that's meiosis. Now, meiosis is something that's not to be studied in detail in your syllabus. Hence, I'm not going to be going over the concept in detail. Just giving you an idea of exactly what meiosis is. Meiosis only happens in one certain area of the body, and that is in the gametes, right? Your egg cells, your sperm cells. That's where meiosis is going to happen. Only in the reproductive organs. Everywhere else in your body, you are going to be seeing what? You are going to be seeing mitosis. In meiosis, what happens is, remember how I was emphasizing in mitosis, the number of chromosomes remain constant. That's not true for meiosis. In meiosis, the number of chromosomes become half, meaning that we go from a diploid. Diploid was when you had two pair of chromosomes, right? So you go from being diploid to being haploid. You go from being diploid to being haploid, only having half the number of chromosomes. That is meiosis. So in meiosis, the number of chromosomes are reduced by half. Why though? Why does that need to happen? Why can't the body just use mitosis again? Because in sexual reproduction, we have got two gametes. We have got the male gametes and we have got the female gametes, right? Now, the male gamete will have 23 chromosomes. The female gamete will also have 23 chromosomes. So, when fertilization is going to occur, the 46 chromosomes will be restored, right? 23 from the male, 23 from the female. Combine them together, you get 46, right? If both the male and the female gamete had 46 and 46 chromosomes, so when you combine them, you would get a huge number of chromosomes. That's not how the body works, right? For it to be a human, it has to be 46 chromosomes. So in meiosis, the number of chromosomes is half. So meiosis occurs only in the reproductive organs, only in the gametes. Everywhere else, you're going to be dealing with mitosis. Other than this, you would other than this, you do not have to know anything else other than this regarding meiosis. So This is how the cell actually divides, right? But when we talk about the cell is dividing, fertilization has happened. Now you have a zygote, right? That's what you get. When fertilization happens, you have a zygote. So when you're going to have a zygote, what is going to happen? That now this fertilization has happened. So the cell will now divide by mitosis. Okay. So even after fertilization, when the cell is formed, you will divide by mitosis. Okay. So that is what you guys need to know. Now, let me know if there's any confusion in what we have discussed so far. Next, we're going to go ahead and take a look at how inheritance happens. So how does the actual portion of inheritance happens? So we're going to go ahead and take a look at that. Does anybody have any question meanwhile? Would anybody like to ask anything?
Miss, are we gonna go over the genetic diagram?
Yes, we are going to go over the genetic diagram, inshallah. Okay. So if this is fine, let's go ahead and go over our inheritance. Okay, so when we talk about inheritance, right? What actually is inheritance? Inheritance is basically the passing down of genetic information from one generation to another. So if I'm passing down genetic information, if I'm carrying down the genetic information from one generation to another, that is what we call as our inheritance. Now, before we can understand how the genes actually pass down, how does inheritance actually occur, we have to understand a few terminologies, right? First of all, let's go ahead and talk about what is an allele. Okay. When we talk about an allele is an alternate form of the same gene. What does that mean? Okay. So let's talk about, for example, eye color. Okay. In eye color, the gene is for color of the eye. That is not going to change. The gene is for the color of the eye. That is not going to change. It's going to be the same gene, right? The gene is going to be for the color of the eye. We're not going to be talking about anything else. Gene is for the color of the eye. Now, in the color of the eye, we can either have a blue color or we can have a hazel color or a brown color or any other possibility. Black color, so many possible combinations are there, right? So that is the same gene. It's the gene for eye color, but we have a different form of it. One is brown, one is black. That's what we call an allele. Then, when we talk about another terminology that you need to know is locus. What is locus? Locus is the location of a particular gene on a chromosome. We geneticists, right, know know exactly what gene is located on what portion of the chromosome. And this is exactly, if you guys ever read any research articles, that's exactly how you guys are going to be seeing the information that they will write down. This was the locus, this arm of the chromosome, this number of the chromosome, and then this region of the chromosome. So every gene has a particular location on the chromosome. You can find it there every single time. You can find it there every single time. So every gene is going to have its own particular locus. Then another terminology that we have to understand is genotype. What is genotype? Genotype is the genetic makeup of an organism the genetic makeup of an organism. So if I am talking to you guys, right? We are scientists, we're doing research work. I say, listen, when we're going to go ahead and talk about it, this is the genomic information. This is the DNA. This is what the DNA says. What the DNA says is the genotype. But you and I both know that whatever the DNA says is not necessarily what we see. The DNA can say, for example, this person has a blue color. But when we look at the actual person, their eye color might be black, right? So the observable features of an organism are phenotypes. So genotype is what's actually present on the DNA level. But phenotype are the features that I am actually able to observe in an organism. Now, we just discussed alleles, right? That every gene can have an alternate form. When we talk about alleles, so alleles can be of two types. The gene can have different forms. Either we can have the dominant allele or we can have the recessive allele. The dominant allele is the allele that will express itself even if there is only one copy. What does that mean? That even if you have, for example, let's say that a person has two alleles for eye color. The person has one allele for black and one for blue. The black one is dominant, meaning that you will see black color even if there is only one allele, even if there's one black, even if there's just one single black allele, but that's what you're going to see because it's dominant. So it's going to overcome the effects of the other one. Then we can have recessive allele, right? In recessive allele is the allele that will only express means show, that we can see it on the person, will only express itself if there are two copies of it. So the person, if the person is supposed to get blue eye color, it will only be visible, it's only going to show up if he has both blue color alleles. Okay, so if both of the alleles are blue, the person is going to show us blue colored eyes. If he only has one allele for blue, we won't see anything. So it's a recessive allele, it does not show up easily. Now, when we talk about our alleles, right? By the way, this is something you guys are studying at a much simpler level, right? When we talk about alleles, we only talk about two alleles. For example, I'm talking about eye color, I'm talking about black allele and blue allele. A lot of your, by the way, genetics is not that simple. Genetics is a very complicated topic. A lot of these features that we inherit are actually affected by multiple genes. We're studying it at a very simple and a very, in fact, quite a simplified level. So when we go ahead and we are going to talk about it, so one allele is going to be inherited from both of the parents. Okay. What does that mean? That means that we're talking about eye color. Let's continue by talking about eye color. We have two parents. Okay. Both the parents, the father, let's say, has black color eyes. Okay. So the capital B means black. Okay. Black colored eyes. Small b means that you have blue color eyes. Now, let's say over here, there are three possible combinations. Either the person can have both the alleles are black. Okay. If both the alleles are black, what term do we use? We will say that the person is homozygous dominant. Homozygous means that both the chromosomes are the same. Okay, homozygous means that both the chromosomes are the same. So in this case, both the chromosomes are the same and both of them are dominant. So homozygous dominant. Then the second possibility is that both the alleles are for the blue eye color, in which case we will say that the person is homozygous recessive. Homozygous means same. Recessive means that they are both the recessive genes. And the last possibilities that we can have is that we can have one dominant and one recessive. So if you have one dominant and you have one recessive, that will be known as heterozygous. Okay. Now, let's consider multiple possibilities. We will consider all of the possibilities. Okay. Let's say that we have a father who has a black eye color. Okay. The father has a black eye color. So this is the genotype. Okay, the genotype is going to be capital B, capital T. The person has a black eye color. He is homozygous dominant. Okay, then we have our mother. The mother has a blue eye color. So obviously, if she's blue, she has to be recessive. She has to be homozygous recessive. Because if she was not homozygous recessive, even if she had one allele of the black color, she would not be able to see. I mean, she would not be able to have the blue color, right? Now, let's make our gametes. So, the gametes for the father, they all will have the black eye color gene. And for the mother, they will all have the blue eye color gene. Now, you're going to make your genetic cross. The genetic cross is your monohybrid cross. This is how we make the genetic cross. First, I'll write down the information of the father. So father has a capital B and a capital B. Then we write down the information of the mother. The mother has a small b and a small b. Now, the offspring is going to get one allele from either parent. So, they will get a capital B from the father and a small b from the mother. A capital B from the father, small b from the mother. Capital B from the father, small b from the mother, and capital B from the father and small b from the father. So all of the children are heterozygous, all of them. And even though they are heterozygous, even though they have the allele for blue color, but they will all of them are going to have black eyes, right? Because the black allele is the dominant one. So all of them are going to have black eyes. Now, let's consider the same scenario. The father has black eyes, but the mother has blue eyes. This time though, the father's black eyes are heterozygous. Okay, so he has one dominant and one recessive. So when you will make your genetic cross, it's going to look something like this. The child will get the capital B from the father and the small b from the mother. In the second block, you can see that I'm getting small b from both the parents. In the third block, capital B from the father, small b from the mother. And in the last block, we will have small b again. In this situation, now what are we getting? We are getting a 1:1 ratio, where half of the kids are going to have a blue eye color and half of the kids are going to have a black eye color. 1:1 ratio, or 50%, 50%. 50% of the kids are going to have black eyes and 50% of the kids are going to have blue eyes. Now, what happens if both of the parents have black eyes, but both of them are heterozygous? Right? So in this case, you're going to solve the question in exactly the same way as I just told you. Like this. The alleles for the father, you can write on top. Alleles for the mother, you can write on the side. The capital B, capital B, capital B, small b, capital B, small b, and small b, small b. So in this case, what is the ratio we are getting? 3:1. 3 of the kids will have black eyes, and one of them is going to have blue eyes. So either you can write down 3:1, or you can write down as 75% and 25%. So 25% chance that the child is going to have blue eyes. Otherwise, there's a 75% chance that the child is only going to have black eyes. So this is how the genetic cross is made. This is how the genetic cross is actually formed. Right? I hope this is making sense. If anybody has any questions, if anybody would like to ask anything, let me know. And if there are no confusions, then we can go ahead and we can take a look at the next portion that we have, and that is all right, I'll explain to you the hetero and homo portion once again. Okay, so think of it this way. Like I said, we have alleles. Okay, in alleles, what happens that we can have an alternate form of the same gene. What is the gene? My gene is color of the eye. That's my gene. Okay, so I need to have I will, in the color of the eye, I'm going to continue with the example we were discussing. We can either have a black color or we can have a blue color. Now, every person has two alleles. Okay, every person will have two alleles. So it's not that I can only have the black allele and that's it. The other one is empty. No, we always have two alleles, right? Why? Because all the chromosomes are in pair. If there is an X chromosome, there's a Y chromosome. If there is chromosome number 1, then there is another copy or another pair of chromosome number 1. Okay? So if on one chromosome, right? So if you have the black, right? Then you will have an allele for it. That's going to be the blue. Okay? So what is going to happen that for the color of the eye, you're not just going to have one option. The person is always going to have two alleles. Okay? Now, in the two alleles, there can be multiple combinations. Either both of the alleles can be black. Okay? If both of the alleles are for black color, that is homozygous dominant. Why? Because both the colors are the same. Both of them are black. So homo generally means same. Okay. So both of them are the same, right? That's homozygous dominant. Then if the person has one black but one blue. So if they have one black and one blue, that is going to be known as your heterozygous, because the person now has different chromosomes. So this is going to be heterozygous. And if the person has, both possibilities are that the person is having blue, so that will be homozygous recessive. All right. So that's what your heterozygous and homozygous mean. Okay. Now, the next portion that we have to go ahead and we have to talk about is the portion that is related to your codominance. Okay. What is codominance? So sometimes what happens is that in codominance, what happens that I told you guys we can have one dominant and one recessive. In codominance, it so happens that both of the alleles are dominant. Let me give you guys an example of this. A very good example of codominance is your blood group. Okay? What's your blood? What is the blood group all about? Okay, let me explain to you guys what your blood group actually is. What do we mean by it? So on your red blood cells, right? On your red blood cells, we have receptors. Okay, we have these receptors. These receptors are very important. Why are these receptors so important? The reason these receptors are so important is that they are sort of the identification markers of the cell. They're like the ID card of the cells. Okay. All our cells, by the way, if we get the time, we'll go over immunity as well. All the cells in our body have these sort of receptors, have these sort of markers. Okay. They're like ID markers. All the cells in our body have them. That's how our body knows if the cell belongs to our body or is it something from the outside. That's how the cell can tell if the that's how the cell can tell. That's what the body tells. Oh, this cell belongs to our body. No, wait, this cell is somewhere from the outside, with the help of the identification markers, with the receptors. So our red blood cells have receptors. Okay, our body can either have receptor A, receptor B, or they can have neither. If they don't have receptor A or receptor B, we could say that they are blood type O. Okay. Now, the blood type, the thing about blood type is that A and B, both of these, they are alleles. How are they alleles? They're coding for the same gene, blood type. Both of them are coding from the same gene. But there can be multiple forms of it, form A or form B. Now, the thing about A and B is that these two are codominant. What does that mean? That means if they are codominant, so I will see the effect of both A and B. So, what does that mean? If someone is blood type AB, blood type AB means that this person has the allele for A and also the allele for B. They have receptor A and they also have receptor B. Okay? That's what blood AB means. This is an example of codominance. This is an example of codominance because the person has two dominant genes, A and B. Both of them are dominant. So the receptors on the red blood cells will be both A and B. You will see both receptors. You will see receptor A as well as receptor B. So in codominance, both of the alleles are visible. You guys go to your flowers, right? You guys are going to see that the flowers can either be, for example, white or red or maybe pink. That pink might be an example of codominance. Maybe the white and the purple are codominant. So when the plant inherited them, it shows you both of the colors, right? That's an example of codominance. Let's say that you have a person who has blood group A. Okay. So this is how we write down codominance. Okay. I is the symbol that we use for blood type. Okay. So I can have blood type A. Now, if a person has blood type A, so let's say that they have one allele A and they have one allele O because O is also an option. But O is an option, but remember that O is recessive. So O will not show up. Okay. So a person has blood type A, and in the blood type A, one allele is for type A and the other allele is for type O. Then we can go ahead and cross it with a person who has blood type B. This person has one allele for blood type B and the other allele for blood type O. Let's make our cross. So see over here, we will get AB. So in AB, both of the alleles are going to be expressed, meaning that when we take a look at the red blood cells, we will see receptors A as well as receptors B. Over here in this, the second box, I will get B and I will get O. Now, In this case, I told you guys O is recessive. So O will not be visible. You will see blood type B only. You are not going to see blood type O. You will see blood type B only. Then third possibility is that we can have A and O. Again, this is not codominance. This is normal inheritance. This is not codominance. This is normal inheritance. The A allele is dominant. The O is not dominant. So the person will only have receptor A. And the last possibility is that the person can get both alleles for blood group O. So when the person is going to be born, they are going to be blood group O. So in this case, see, there's a 25% chance that the person is going to have blood group AB, 25% chance that the person will have blood group B only, 25% chance that the person will only have blood group A, or a 25% chance that the person is going to have blood group O. So over here, the blood group A and B, that's an example of codominance where both A and B will be seen. So when I will take a look at this person's red blood cells, I will see receptor A as well as receptor B. So this is exactly what your codominance is all about. So you can either have normal inheritance that we discussed, or you can have your codominance, right? Anybody has any confusion? Would anybody like to ask anything? Okay, so how do you tell if a disease is sex-linked or not? Good question, Tia. When you are going to go ahead and talk about it, first of all, let's discuss what are sex-linked diseases. Sex-linked diseases are those diseases that are inherited on the sex chromosomes. Okay, so the sex chromosome are the ones that are actually going to carry the allele. Okay, now what happens is that so that's a sex-linked gene. Any gene that is a sex-linked gene, it will be found on the X chromosome. If the gene is on the X chromosome, we will say this is a sex-linked gene. That's how you will know, right? So if it's on the X chromosome, it's a sex-linked gene. Now, you guys must have studied that there are certain diseases that are sex-linked diseases. A really big example of this that I believe is in your syllabus as well is hemophilia. Hemophilia is a disorder where the blood does not clot properly. Okay? It's a disorder in which the blood does not clot properly. Okay? So, what happens in hemophilia is this, that hemophilia is a disease. The gene for the disease is found on the X chromosome. Okay? The X chromosome is going to have the gene for the hemophilia disease. Now, what happens is this, that okay, so when first clue that you will often get in past papers is that you will see that there will be an X written, and on top of the X you will have a G. So that's how you will know that this is a sex-linked disease. Now, what happens in sex-linked diseases? Let's say you have a woman, a mother. That mother is a carrier of the hemophilia gene. Okay, the small H is for hemophilia, the capital H is for a normal blood. So a mother has a gene. She has a recessive gene for hemophilia. The gene for hemophilia is recessive. Now, what is going to happen for it? Let's say that we have a completely healthy male, very normal, does not have any hemophilia. Okay, now this healthy male will have one X chromosome and one Y chromosome. Now, what is going to happen? What's going to happen is this, that when you're going to make your cross, right? So this is possibility number one. This is possibility number two. This is possibility number three. And this is possibility number four. Possibility number one is that you can have a female child that is completely normal. So X, and both the alleles are for normal patch. So this person is going to be completely normal. The second possibility is that you can have a second combination. The second combination that you can go ahead and have is that you can have a girl, but this girl has the recessive gene. Okay, this girl has the hemophilia gene, but the hemophilia gene is recessive. Since the hemophilia gene is recessive, so it will not show up. This person is going to be completely healthy, but we will consider this girl to be a carrier. Carrier meaning that she still has the recessive gene. The recessive gene with her is still present. Okay, she still has it, the recessive gene. That recessive gene is not going to show up. She's not going to get sick, but she can pass it down to the next generation. Then the third possibility is that you can have a child, a male child that will be completely normal. And the last possibility is that you have a child, a male child, but this male child will have hemophilia. Now, you can wonder, I don't know if you guys are wondering this or not, but you can wonder why exactly is this male child getting hemophilia? He only has one copy of the recessive allele. Our teacher, when she defined the recessive allele, she said it should have two copies. If we have two copies of the recessive allele, only then it will give us any, effect. So why is it that with just one copy, I'm seeing hemophilia? Now, if you guys ever take a look at the chromosomes X and Y, you will see that the X chromosome is actually much larger in size than the Y chromosome. Now, what happens is that Y chromosome, so what happens is that since the X chromosome carries more genetic information than the Y chromosome. So what happens that some of the genes are found on the X chromosome, but the Y chromosome has no allele. The Y chromosome has nothing corresponding to it, right? The Y chromosome has nothing. The X chromosome has a gene, but Y chromosome has nothing. The same is true for hemophilia. We can have a gene for hemophilia on the X chromosome, but not on the Y chromosome. Okay? So even if we have a recessive copy coming from the mother, because the Y chromosome has nothing to offer against it. It doesn't have anything to offer. It doesn't even have the gene at all. It has nothing. It doesn't have the gene. So obviously, even if there is just one of it, the person will still get hemophilia. So these sort of diseases, hemophilia, different types of color blindness, these are all considered to be sex-linked diseases because they are found on the X chromosome and they are inherited accordingly.
Miss.
Yes.
So Miss, a male is bound to get hemophilia no matter what?
No, Arham. Say
Or is it a 50-50% chance?
over here, you can also have
Okay.
a normal allele, right? If they
Okay.
get the normal allele, then they are, yeah. So there's a 25% chance. Okay. So technically,
Okay.
there's a 25% chance that you will have a normal female, 25% chance that you are going to have a 25% chance that you're going to have a female that's going to be a carrier, and 25% chance that you will have a normal male child, and a 25% chance that you will have a male child with hemophilia.
Okay.
All right. Okay. Perfect.
Yeah, thank you.
So that's what we need to know regarding our inheritance. Now we're going to go ahead and we will take a look at another important portion of the DNA, and that is how your proteins are made. Give me 1 second. I'm going to share the screen with you guys. Again, hold on. Let me know meanwhile if anybody has any questions, and if there's any topic you guys want to go over as well, you guys can let me know. And if we have the time, I'll definitely go with it. Okay, all right. So if there are no questions from anyone, let's go ahead and go over our portion related to our protein expression. Okay, so when we go ahead and we talk about protein formation, how is protein formed? Protein is obviously formed with the help of a DNA, because DNA is the one that has all the genetic information. The DNA is the one that has all the genetic code that we need for any protein to be made. For our example, we are going to go ahead and we're going to talk about insulin. Okay, that will make things easier for us. Now, for me to make any proteins, there are two steps that you have to do. I will go over the steps in a simplified manner with you guys. When you guys, inshallah, do your A levels, you guys are going to study this in a lot more detail. So, we have two steps. You have transcription and you have translation. I will tell you, first of all, let me just quickly tell you what these two steps are, a summary of these two steps. In transcription, you will take your DNA and you will use it to make something that we call messenger RNA. I will explain what that is. Okay. In translation, you take the messenger RNA and you use it to make a protein. Now, what happens is this. On your DNA, you have your genetic sequence, right? So we have a strand of DNA over here. This is the genetic sequence found on the strand. Okay. Let's say that this is the sequence for making insulin. What is going to happen? That the strand is going to open up. After the strand is going to open up, now I need to somehow take this genetic information and use it to make a protein. To do that, I have to take out this information from the nucleus, out of the nucleus. If I want to make a protein, I somehow have to take this information and carry it out into the cytoplasm. Let me give you guys a good analogy, okay? When we're going to go ahead and talk about it, think of it this way, that you have an office. Inside that office, you have piles and piles and piles and piles and piles of information. You want to use that information to make a machine. You go into the office, what are you going to do? You're going to take out the paper that you need that has the information regarding your machine. Then you will copy that information down on another paper. After copying that information down on another paper, you go out of the office, you go to your workshop, your lab, and you start making your machine. That's what is happening over here. The DNA has the genetic information, but it can't leave the nucleus. So all of that information will be copied onto the messenger RNA. The messenger RNA will carry all of that information out of the nucleus to the cytoplasm where the protein will be made. That's the basic concept of it. Okay? Now, what actually happens, again, I'm going over the basics. You guys are not required to know this in too much detail. What happens is that we are going to use, so the DNA will open up. Okay, the DNA will open up and it will say, listen, this is where the code for insulin is. So you need to start, copying the information down. That information will be copied in the form of messenger RNA. How do we make the messenger RNA? So messenger RNA is actually going to be complementary to the DNA. So it's going, how you have your nucleotides? Just like that, your nucleotides are going to start to line up in front of the DNA. Just like you have your nucleotides in DNA, so RNA also has nucleotides in it. Those nucleotides are going to start to line up in front of the DNA. But remember this, that the base pairing is always complementary. Even if it's DNA or RNA, doesn't matter. So in front of thymine, we will get adenine. In front of cytosine, we will get guanine. In front of guanine, we will get a cytosine, right? So complementary nucleotides will start to arrange themselves in front of the DNA, and they will copy that information down in the form of messenger RNA. Now, there's one small difference between DNA and messenger RNA. In fact, there are few differences. One major difference between your DNA and RNA is DNA has 4 bases that I told you guys: adenine, guanine, cytosine, thymine. RNA also has 4 bases: adenine, guanine, cytosine, uracil. It does not have any thymine. RNA does not have any thymine inside it. RNA has uracil. RNA does not have any thymine. RNA has uracil. Okay. So instead of T, we have U. It's a replacement. So in the RNA, the adenine will not make a bond with thymine. It will make a bond with uracil because RNA does not have any uracil. Sorry, it does not have any thymine. It has uracil. That's the first difference between DNA and RNA. Then, another difference between DNA and RNA is that DNA is made up of deoxyribose sugar. RNA is simply made up of ribose sugar, the normal ribose sugar. DNA is made up of deoxyribose that has less oxygen. RNA is made up of normal ribose sugar that may be present, right? Then, if we continue, another difference between RNA and DNA is that RNA is a double-stranded structure. RNA is a single-stranded structure. So there's only one strand of RNA. So all of the information from the DNA is going to be copied onto the RNA. The RNA is going to have the complementary base pair. So whatever is on the DNA, the RNA will get the complementary base pair and the RNA will be made. Once the RNA is made, now it has all the information it needs to make the protein. So that RNA will leave the nucleus and then it will go into the cytoplasm. Okay. Now that the RNA is in the cytoplasm, the process of protein synthesis can begin. To make proteins, now that the RNA is in the cytoplasm, to make proteins, I require 2 pieces of technology. Not technology, 2 pieces of 2 types of molecules. You require ribosomes, which I'm sure you guys must have studied, right? Ribosomes are responsible for protein synthesis. So I need ribosome. Along with ribosomes, I need another molecule known as transfer RNA. Transfer RNA is also an RNA, just like the one we studied, but it's slightly different. It's a bit different. Okay. So we have transfer RNA. Okay. Transfer RNA and ribosomes together, they will work together to make the protein. Now, on our RNA, on my RNA, I have something known as the triplet code. What is the triplet code? The triplet code is if I take 3 nitrogenous bases, CAC, and I look at them together, so that 3 of the nitrogenous bases is one triplet code. Then the next 3, for example, in this case, CCC, are the next triplet code. GUA is the next triplet code, right? So 3 of the nitrogenous bases is equal to one triplet code. And one triplet code is going to be, one triplet code is specific for one type of amino acid. Right? So for example, if there is CAC is one code. So the body knows CAC is the code for amino acid glycine. Okay, so wherever I see CAC, we have to insert glycine amino acid. The next one, CCC. Okay, CCC is the code for leucine. We have to insert leucine amino acid over here, right? So the messenger RNA has something known as triplet code. Triplet code, think of it like a shortcut, right? It's a shortcut. It's an abbreviation. The body reads the abbreviation and it says, okay, This is the code for this amino acid. So the body knows that this is the amino acid they have to insert. So we have triplet code. Okay, triplet code is going to be 3 nitrogenous bases. Now, what is going to happen that the messenger RNA is going to come to the cytoplasm. When it's going to come to the cytoplasm, it will bind to the ribosome. The ribosome is responsible for reading the RNA. It reads the RNA. It's like, okay, what's written? What's written? And as it is going to read the RNA, what is going to happen? Now, coming back to transfer RNA. A transfer RNA has two important parts. A transfer RNA has something that we call anticodon. What is anticodon? Anticodon is the opposite of triplet code. Okay. So for example, if I have CAC, so they will have the opposite of it. They will have GUG, right? The opposite of CAC is GUG. Okay. Now, it will have an anticodon, and along with it, it will have a area where it can attach to the amino acid. So what is going to happen that the amino acid starts, the ribosome starts to read the messenger RNA. It says, okay, there is CAC written. So whichever transfer RNA that has the anticodon, whichever transfer RNA that has the anticodon that matches, that is opposite to CSC, that transfer RNA will come and it will bring the matching amino acid with it. Whatever CSC is coding for, whatever amino acid it is coding for, it will bring that amino acid with it. Right? Then the ribosome will move on to the next part. It will say, oh, we have AUG next. So can the transfer RNA that has the matching codon, can the transfer RNA come? So whichever transfer RNA has the matching codon, that transfer RNA will come. And it will bring with it the amino acid that matches the code. Peptide bond will form between the two amino acids and the transfer RNA will go away. They have done their job, they've got the amino acids, they will go away. The ribosome will continue reading the messenger RNA, and one by one, they will keep telling the transfer RNA, one by one, the transfer RNA that matches the sequence is going to attach. It's going to bring the amino acid that is required. The amino acids will keep making peptide bond, and eventually you will have a whole protein ready. So that is how your protein synthesis occurs. So step number 1, open your DNA, open wherever you need to, whatever protein you need to make the RNA. The RNA is going to be complementary to the DNA. Okay? Now, the RNA will leave the nucleus and go into the cytoplasm. The RNA has the triplet code on it. The triplet code, every code is specific for a certain amino acid, right? So if CAC is present, that is only for glycine, nothing else. The messenger RNA will be read by the ribosome. The ribosome is going to read the messenger RNA. And as it is going to read the messenger RNA, it's going to bring the corresponding transfer RNA. So whichever transfer RNA has the matching code, that transfer RNA will come and it will bring the matching amino acid with it. The amino acids will join up with each other with the help of peptide bond and proteins. That's how proteins are made, right? Is this making sense? Is there any question that anybody wants to ask?
So miss, the tRNA holds like three nitrogenous bases for one molecule and one
For one,
amino acid.
yes. Yes,
Okay.
yes. The anticodon has 3 nitrogenous bases, yes.
Okay. That's it. Thank you.
Okay, all right. Okay, so if there are no other questions related to our protein synthesis, yes, Rani, we will in a little bit. Okay, so um Okay, so some of you wanted to go over uh, uh, the hormones that act during fertilization, and your hormones that are FSH, LH, and estrogen. So yeah, we'll go ahead. We'll take a look at that as well. Okay,
Miss,
so
can we go over the eye?
yeah, if we get the time, inshallah, we'll definitely go with that as well. Okay. So, in fact, we'll go over it. Don't worry. We'll go over it. And if there are any other topics you guys want me to cover, by the way, go ahead, list them out in the chat so I know what you guys want, topics you want me to do, like topics you guys want me to do. So I'll concentrate them on them more than the other ones, right? Okay, so some of you wanted to go over the topic of reproduction, especially the hormonal portion of it. So we'll go ahead and we'll take a look at that. Anybody else, any other topics you guys want me to cover, please list them down. It makes things easy because then I can make sure that I get the topics that you guys are actually confused about.
Miss,
Yes, Sarham?
making of yogurt. Uh,
Okay. Okay. All right. Okay. So another, yeah, we'll go over biotechnology as well. I'll try my best to cover kidney as well. Okay. In fact, that's what I was going to do next with you guys. I was going to go ahead and I was going to go over biotechnology. So let's go ahead and take a look at that. 1 second. Let me share the screen with you. So when we are talking about our biotechnology, right? There are few topics in biotechnology and genetic engineering that I feel like are really important for you guys to know. So, what are the topics that you guys need to actually go ahead and you guys need to know? So, one of the applications that we have to go ahead and we have to talk about is genetic engineering. Okay, we have to talk about our genetic engineering and we have to talk about how exactly is the genetic engineering done and how exactly a big example of genetic engineering is how the body makes insulin. So, to understand how genetic engineering happens, how is it that I can use genes to, for example, in the case of insulin, I take the bacteria and I use those bacteria to make insulin. That's one example of genetic engineering. Another example of genetic engineering is when I, for example, I want to go ahead and I want to get a crop of some uh, pesticide resistant tomatoes. So what I'm doing is that I'm inserting the gene for the pesticide resistance in my plants. That is genetic engineering, right? So in genetic engineering, what are we actually doing? We are taking a gene and we are inserting it into another species DNA. I'm inserting it into another species DNA. That is what we call as our genetic engineering. Okay. Now, in our genetic engineering, when we are going to talk about it, a big example of this is how we get the bacteria to make insulin. At one point in time, we did not have the insulin that we have now. We used to extract insulin from animals as treatment for diabetes, but that usually came with a lot of drawbacks, right? The drawbacks that it came with are that one, obviously, you are inserting something that is foreign. It is something that's outside the body. So when it was injected in the person, it tended to often cause allergic reactions. It would cause, the immunity to act up. So that was a problem with it. Then there was some ethical considerations. Obviously, the ethics of taking something from animals and injecting it into humans. So that was a problem that was present until the scientists managed to start producing human insulin with the help of bacteria. And how did they do that? They did it with the help of genetic engineering. Let's talk about how this is done. This is not just for insulin. We use it for a lot of chemicals. We use it for a lot of different techniques. Like I gave you the example, I want to make my plants resistant to herbicides. I'm going to use genetic engineering. Okay. So how do we actually go about it? We will use the example of insulin to understand it. First of all, we often use bacteria for the help of genetic engineering. Why bacteria? Why am I putting the gene in the bacteria and not like in a virus? Or forget a virus, a fungus, or in some animal. There are multiple reasons why these are the choice. These are our choice. We choose bacteria. The reason for that is number one, because bacteria they can reproduce very, very, very quickly. The bacterial numbers, they tend to double like this, like with the snap of your finger, um, the bacteria can actually divide. So it's really easy to obviously make large quantities of insulin using bacteria. Secondly, bacteria tend to form proteins that are quite similar to ours, right? They are quite similar to our proteins. So when if we insert the gene into bacteria, there is a very good chance that they are going to be able to synthesize those proteins because the bacterial proteins tend to be similar to us. Then, when we talk about our bacteria, so obviously bacteria are easier to handle, they're easier to culture, they're easier to grow. So bacteria are the choice. Now, what we normally do is that I will take my gene of choice and I will insert it inside the bacteria. If you guys remember your topic of cell, you must have studied about eukaryotes and prokaryotes, right? Your prokaryotes, especially bacteria, right? When we talk about the structure of a bacteria. So a bacteria has a, one second. So a bacteria has a central loop of DNA. Okay, so in the very center of the cell, it tends to have all of the DNA put together like this. Now, along with all of the DNA that has been put together, we also have some extra genetic information. That extra genetic information is found in the form of a small loop of DNA that we call plasmid. So this plasmid that has extra genetic information, any extra information, for example, if the bacteria develops a mutation, it's probably going to be on the plasmid. That is all found in the plasmid of the bacteria. So when I will start the process of genetic engineering, what am I going to do? First thing is first, I need my plasmids. Then, along with my plasmid, another reactant or another ingredient that I need is I need my human DNA, right? So for example, I want to make insulin. If I want to make insulin, so I need the gene for insulin. This is my DNA. So I need to get my insulin gene. Now, I need a way of cutting out the insulin gene. How do I cut out the insulin gene from the DNA? I do this with the help of some really special enzymes. These enzymes are known as restriction enzymes. The restriction enzymes are the ones that are going to be responsible for cutting the DNA. They are going to cut the DNA and they're going to take out the gene. So whatever gene I need, they're going to go ahead and they're going to cut it. Along with that, I also need to cut the plasmid, right? Why do I need to cut the plasmid? Because I need to make space. How am I going to put the gene in there? So I need to go ahead and I need to make a cut in the plasmid. For that, I will use the same restriction enzyme. So remember this, both the gene of interest, the gene that I want to insert, my insulin gene, and the plasmid are going to be cut with the same restriction enzyme. Same. Okay, we cannot use different restriction enzymes. They have to be the same. Now, what does the restriction enzyme do? The restriction enzyme will cut the plasmid and the DNA at a specific area. That area is decided by us. We are geneticists. We have studied the sequence. We know where everything is. We design them. Okay? They will cut the gene exactly where we want them. And what they're going to do is that, so the ends of the plasmid and the ends of the DNA, right? These ends of the plasmid and the ends of the DNA that have been cut by restriction enzymes. These cut regions, we call them sticky ends. Okay, so both my DNA as well as my plasmid will have sticky ends. These sticky ends are basically just the cut region. So wherever I will cut my plasmid and my gene, so the ends that will be present, those ends are known as sticky ends. Now, the sticky ends of the DNA, the sticky ends of the gene, the sticky ends of the insulin gene are complementary to the sticky ends of the plasmid. Okay? These are complementary. These are complementary. That's why I'm using the same enzymes, because I want the ends to match up. If the ends will match up, I will see successful insertion. So I want the ends to match up. So these sticky ends are complementary to each other. The sticky ends on the plasmid and the sticky ends on the DNA. Now, what is going to end up happening? That because the sticky ends are matching up, they're complementary to each other. So this you can say your DNA, your insulin will manage to successfully insert itself into the plasmid because the ends were complementary, right? Now what has happened is that the plasmid has the gene for insulin. I am going to insert the plasmid back into the bacteria, right? Normally, what do? We just add the plasmid to the bacteria, wherever the bacteria are, wherever we are growing them, we just add the plasmid. Okay. So what is going to happen? That we will add the plasmid to it. And once the plasmid has entered the bacteria, we allow the bacteria to grow. The bacteria grow, they divide, and as they are going to be producing proteins, as they're going to be doing protein synthesis, so they're going to be doing protein synthesis of the entire plasmid. So they will be making their own proteins, but they will also be making the insulin protein for us. So that's going to go ahead and that is going to help us out and actually lead to the formation of your insulin gene, right? So same concept is used for multiple things. If I want to make my tomatoes resistant to pesticides, I can take the resistant gene, put it inside the tomato gene. Okay, same concept. So this is one of the principles of genetic engineering. Genetic engineering is a very vast topic. If any of you decide to take it up to a graduate level, you guys are really going to enjoy it. It's a very interesting topic and it's got numerous applications. But this is one of the major things that is mentioned in your syllabus, which is how genetic engineering is done. So these are the steps you should know, that you will take a plasmid and we will take the gene. We will cut both of them with the same restriction enzyme. The result is going to lead to the formation of sticky ends. The sticky ends of the gene and the plasmids are complementary to each other. So the sticky end will insert into the gene. Okay, one point that I wanted to add. After I have inserted the gene into the plasmid, I will join it together. And I will join it together by using an enzyme known as DNA ligase. So we will go ahead, we will insert our gene of interest using DNA ligase. And after that, what are we going to go ahead and do? We are going to seal the sticky ends. We will join the sticky ends together using DNA ligase. Put the plasmid with the new gene back in the bacteria, let them grow, and as they grow, they will start to produce insulin for us. Making sense? Would anybody like to ask anything?
Yes, miss. Like, are there any other methods of like making like genetically modifying an organism? Like, I think viruses are also a way to modify the DNA.
Viruses, the problem, yeah, we can. The problem with viruses is that viruses, when you go ahead and talk about it, they are non-living, technically they're not classified as living organisms, right?
Yeah.
Because they cannot reproduce on their own, they cannot make their own proteins, they cannot make their own they cannot do anything pretty much without the help of the host cell. So if you do decide to use a virus, now with the virus there are a few problems, right? The problem
Okay.
is that inside the virus the genetic code is so some viruses, for example, they do not even have DNA, they have RNA. So we can have RNA viruses, right? So inserting DNA into an RNA is going to be something that is tricky. Then when you talk about your viruses, so viruses technically will not be making the protein themselves. I cannot put the insulin gene in the virus and they'll produce the gene by themselves, right? I will have to put them in some other host cell. And the way viruses work is that viruses sabotage the host body. So they literally take over the host cell. So if a virus gets in my cell, what is it going to do? It's going to take over my cell and it's going to start using my cell's organelles, the machinery inside my cell to make its own proteins. Okay, so viruses are not as commonly used as bacteria. Bacteria is one of the ideal choices we have at the moment because viruses will not make the protein themselves unless you put them inside some kind of host. And viruses are much smaller in size as well. And they're not all viruses have DNA, some of them have RNA. So we generally, viruses are not used very commonly. DNA is the one, bacteria is the one that we prefer. Bacteria are closer to us, especially when it comes to making, human products. Okay, all right. Okay, guys, so we're going to go ahead. We'll take a break over here. We'll take a 15-minute break. And after that, we'll go ahead and we will resume. Meanwhile, if anybody has any questions or any topics that they want to go over, again, go ahead, send them to me in the chat. And we will start, go ahead and we will go ahead and we will I'll try to go over them, you guys, as much as possible. I'll go ahead and I'll try to go over them. I wish we had more time, or I wish that I had you guys join earlier. We could have gone over these topics in proper detail and studied them. Well, regardless, we are starting the classes for A level soon, inshallah. So I'm hoping that I'm going to see you guys again in my classes. We do have limited seats. We only take 3 students per batch. We don't take more than them. So please do go ahead and sign up if you are interested. So we'll go ahead, guys. We'll take a 15-minute break and we will resume after 15 minutes. Okay. So, all right. So I was just going over the topics that you guys listed in the chat. So, kidney, inspiration and expiration. Someone wants to go over. You want to go over the hormones that act during fertilization, and I think someone also wanted to go over the topic of eye, right? Okay, so we will go ahead. We'll go over all of these topics. If anybody wants to me to go through anything else, do let me know. Yes, I'll go over that as well. Okay, so let's go ahead and take a look at the topic of kidney. Give me 1 second. All right, so going over our kidney, right? In our kidney, when we are going to go ahead and talk about it, so the kidney obviously is responsible for something that we call excretion. When it comes to excretion, so excretion is not a function specific to the kidney, right? Excretion is simply the removal of waste product, toxic materials, toxic substances, waste substances from the body. Your body removes waste substances through your skin, your sweat glands. They remove waste products through your respiratory system, carbon dioxide. Right? Carbon dioxide is a waste product and it's removed with the help of respiration. And of course, then we have another major organ that's your kidney. So, when we talk about your kidney, how does the kidney actually work? For us to understand how the kidney work, let's go over the basic structure of a kidney. This is the structure of a kidney that we see within the body. And then we will also talk about the structure specific to the kidney itself. Your urinary system generally obviously consists of your kidney. We have a right kidney and we have a left. The right kidney and the left kidney are connected to the bladder with the help of a ureter. Your ureter is a tube that carries the urine from the right kidney or the left kidney to your urinary bladder. The urine is going to be stored inside your urinary bladder, and then the urine opens up, or sorry, the bladder opens up to a small tube that we call urethra. And the waste products are expelled through the urethra from the human body. Now, if we talk about the structure inside the kidney, right? Like if I take a kidney and I cut it up, what will I see? Well, if you take a kidney and you cut it up, you'll see something like this. You will see that the outer region of the kidney happens to have sort of a dark color. That dark color region of the kidney is known as the cortex. Then, the inner region of the kidney is going to have a lighter color. That lighter color is known as the medulla. Then, you will see that all of the medulla, right? All of the medulla are arranged around a central region. This central region is what we call as the pelvis of the kidney. And then this central region continues down to make a tube that we call the ureter. This is the, you can say, sort of a overall structure of what we see inside a kidney. But if you guys decide to go ahead, take out your microscope and take a look at the kidney, you will realize that the kidney is made up of really small structures that we call nephrons. What are they called? They are called nephrons. This is why, by the way, a doctor who's a kidney specialist, we call him a nephrologist, because the kidney is made up of really small blocks of Lego that we call nephrons. Now, the nephron, the parts of a nephron, you guys need to have an idea regarding that. The parts of a nephron look something like this. We have a small, you can say, capsule, okay? A small bag. This small bag is known as a Bowman's capsule. Then what happens is this, that the Bowman's capsule continues on to go ahead and give you your, one second, let me draw this. To give you a loop-like structure. This loop-like structure that we see, this loop-like structure is known as the loop of Henle. Then, the loop of Henle continues on. The nephron, it wraps around, it continues moving, and then eventually it ends up forming a collecting duct. Okay. Now, inside the Bowman's capsule, we have a bunch of capillaries, a bunch of capillaries. A bunch of your capillaries are all sort of wrapped around and tangled up and just found within the Bowman's capsule. So this is one nephron. A nephron has a Bowman's capsule. That's where you find all the capillaries. Then the Bowman capsule continues on to make a loop, which we call loop of Henle. And then then the loop of Henle continues on and on and on until it goes ahead and gives you your joints together to give you a collecting duct. And that collecting duct is basically going to pass on the waste products, the urine, to your ureter, and the ureter will carry it to the kidney, to the bladder, sorry, and it's going to be removed from the body. Okay, so this is what a nephron is. Now, when we talk about our nephron, so how does the actual process of excretion work? How is it that the body actually goes ahead and it removes the waste products? Because let's be honest, not everything in the blood has to be removed. There are some things that we want to keep a hold of. So how does the body actually do it? The body does it with the help of two processes, okay? The two processes that we have are ultrafiltration. One is ultrafiltration. And the other one is selective resorption. Right? Now, what happens in ultrafiltration? Okay, ultrafiltration happens in the Bowman's capsule. Inside the Bowman's capsule, I have like a bunch of capillaries all, condensed together. Now, what's going to happen that the blood that is coming in all, so this is my Bowman's capsule actually. The blood that is coming in all of these capillaries is under really high pressure. And obviously, you guys know that the capillaries have really small lumen, right? The lumen is really, really, really small. So what's going to happen that as the blood is going to pass through these really, 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 really small capillaries, all of the fluid, everything, all of the fluid is going to move out of the capillaries and into into the Bowman's capsule. Everything except for few. What are the few things that are not going to be removed? Large molecules like large proteins. Okay. Linking this information to the topic of heart, to the topic of capillaries. If you guys remember, you must have studied that capillaries are partially permeable. The wall of the capillaries have small gaps which allow the white blood cells to move out. It allows tissue fluid to move out. So these gaps are present. But these gaps are too small and will not allow any of the plasma proteins, albumin, all those plasma proteins that are found within the blood, they are not going to be able to move out of the capillaries. But other than that, your urea, your water, your salts, sodium, chloride, potassium, your glucose, everything is going to move out into the Bowman's capsule. Hence, this is known as ultrafiltration because this is like the extreme filtration. We are removing everything except for the very large substances like your proteins, like your cells. I will not let my red blood cells leave, but I'm not going to let my proteins leave. But everything else, water, urea, hormones that have been used up, drugs that the body no longer needs, any toxic substances, anything removed. So ultrafiltration. After the ultrafiltration is done, now what has happened is that the body has pretty much taken out everything. The body is like, listen, we're gonna here's some glucose, here's some water, here's the salts that we have, but I don't want to get rid of everything, right? I don't want to get rid of the glucose. I don't want to get rid of the sodium and the chlorine and everything. I only want to get rid of things that I don't need. But in ultrafiltration, pretty much everything has been taken out and given to the nephron. So, step number 2, take it back. We will take back what I need. So whatever I don't want the kidney to excrete, whatever I don't want the kidney to get rid of, I'm going to go ahead and I'm going to absorb that back, right? How am I going to absorb it back? I'm going to absorb it back. So generally, we have this loop of Henle, okay? Now, you guys are not expected to know details of how the loop of Henle works or how the process works. If you guys, again, in A levels, you guys will might cover this, but you guys are not supposed to know, worry about any details about how the process happens. But in selective absorption, what happens is that I will take whatever substances the body needs. The body needs glucose. I will take it back. The body needs water. Let's say that the person's body does not have enough water. It will be absorbed back into the blood, right? This entire loop of Henle, it is surrounded by capillaries. This entire loop of Henle, it's surrounded by a network of capillaries. So whatever the body requires, it will absorb it back into the blood. It will move it from the nephron back into the blood. So glucose is absorbed back. Water is absorbed back. Your salts, sodium, chloride, all of these are normally absorbed back. If they're too much in the body, if you have too much sodium, too much potassium, then that's a different story. Okay. But whatever the body needs for normal functioning, it will take back. Other things like urea, which we don't want, any hormones, any drugs that have been used up, anything that we don't want will stay within the nephron. Anything that the body might need, glucose, any salts that, are needed by the body, water, is all going to be taken back into the blood and returned back to the circulation. So the urine that's going to form is slowly going to become more and more concentrated because you have removed back the water, you remove glucose. So now it's going to have, urea, it's going to have any excess salts, any excess sodium, potassium, chlorine, any excess, drugs, hormones, whatever. And eventually it is going to enter into the collecting duct. The collecting duct is eventually going to pass all of it to the ureter, and the ureter will carry it to the bladder. So those are the 2 steps of how the kidney works. The kidney works, one, by using The kidney works one by using ultrafiltration, in which all of the waste products are basically waste products, glucose, pretty much everything is forced out of the capillaries under high pressure into the Bowman's capsule. And in selective resorption, I will take back whatever my body actually requires. So that is how the kidney goes ahead and does the process of excretion. Anybody like to ask anything about what we have gone over so far? And does anybody have any questions?
Miss, where does the
Miss,
water get absorbed?
Sorry?
Where does the water get absorbed?
The water loop of Henle. Okay. So this area, the loop of Henle, this loop of Henle is surrounded by your capillaries. Okay. So the water moves from the loop of the Henle's back into the capillaries.
but Miss, doesn't the collecting duct absorb water?
The collecting okay, over here the water, the management of water is slightly complicated. The body manages water depending upon how much water is needed by the body. If you do not have enough water in the body, let's say you're fasting, you have not been drinking water all day, your body is going to retain all of the water, meaning that whatever water is going to be present, it's going to absorb all of that back in the blood. On the other hand, if you have excess water in the body, so yes, if you have too much water in the body, then one thing that can happen is that we have something known as protein channels. Okay, water channels. We have something known as water channels. They're literally like a gate, but for water. So the body tends to produce those water channels. And those water channels will allow the water to move into the collecting duct. Yes. But Arham, again, that will only happen if there is too much water in the body. Normally, if you are drinking like just enough water, you're drinking an okay amount of water, the absorption, right, is going to the water is going to be absorbed as needed. It will be taken back into the body. But if there is too much water in the body, then yes, it will pass to the collecting duct, and the collecting duct will
Miss, also one more thing, Miss. What does the loop of Henry absorb?
sorry.
Miss, what substances does the loop of Henry absorb?
Loop of Henle will absorb your glucose. It will absorb
Okay,
back sodium ions, chloride ions, potassium ions, anything else that the body basically requires, it's going to absorb it back. But major thing
okay,
is glucose and your all of your ions, any vitamins that the body needs, any mineral ions that the body needs, they will be absorbed back in the loop of Henle.
okay, Miss. Thank you.
Okay.
Miss, does the water
Yeah.
get collected in the collecting duct?
Yes, the water, if there's excess water in the body, if there's too much water in the body, then yes. So this is something that will happen if there's too much water in the body. So the body does not need that much water. So what the body does is that the body produces water channels. Okay, this is something that's not always present. I mean, it's present, but not always available. Water channels. Okay, so the body activates water channels. Okay, because there's too much water in the blood. So the water channels will allow the water to move into the collecting duct. So the collecting duct will
Normally it
absorb
gets
excess.
absorbed in the loop of Henle, صح? If there's a normal
Yes,
amount of water.
yes, 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 yes. If there's excess water, it goes into the collecting duct. Yes. Okay, all right. And if does anybody have any other question, would anybody like to ask anything else? Okay, so somebody also, okay, you guys wanted to go over, yes, Rani, you wanted to go over the eye, I remember. Okay, we'll go over the eye, and we'll go over the eye, but before we go over the eye, Maham, you wanted to go over the hormones, right? So we'll just quickly take a look at the hormones first, and then we will take a look at the remaining topics. Hold on. All right. So when we are talking about the hormones that are specifically involved in the reproductive cycle, right? So the hormones that are involved in the reproductive cycle are obviously different for males and females. In males, you have testosterone. Mainly, that's the one that's mainly involved and needed for the production of your male gametes. On the other hand, when we are talking about our female hormones, right? So there are four major female hormones that you guys should know about. One is your follicle stimulating hormone, that's FSH. The other one is your luteinizing hormone, that's your LH. So FSH and LH are the two major hormones that you guys should know about. Other than these two, we are going to be talking about obviously progesterone and estrogen. Okay, so four hormones that you guys have to know about. Now, let's go ahead and talk about exactly what these hormones are, how are they produced, where are they produced from, and what is their job in the reproductive cycle. Now, remember this, both follicle stimulating hormone and the luteinizing hormone are produced by the pituitary gland. Pituitary gland is the gland that is, the pituitary gland is the gland that is found inside your brain, right? So inside your brain, there is a small region that actually has the pituitary gland, and that pituitary gland produces a wide variety of hormones. These are just two. That pituitary gland produces so many hormones. It produces your growth hormone, it produces follicle stimulating hormone, luteinizing hormone, it produces your thyroid stimulating hormone. So that pituitary gland has a lot of functions, right? Now, when we are going to go ahead and we are going to talk about it, so these two hormones are produced where? By the pituitary gland. For each hormone, you guys should not only know its role, but you guys should know exactly where it is being produced from. So follicle stimulating hormone and your luteinizing hormone both are produced in the pituitary gland. Then we have progesterone and we have estrogen. Now, I will go over how progesterone and estrogen are made. Estrogen is actually produced by the ovaries. Okay, the ovaries are the ones that are going to be producing your estrogen. Okay, progesterone again is also produced by the ovaries. So FSH, LH from the pituitary gland, progesterone and estrogen from the ovaries. Now, the process that happens is somewhat like this. First of all, you know that inside your ovaries, we have immature egg cells. Okay? We have immature egg cells. Those egg cells need to undergo maturation for them to actually be ready for fertilization. Second. They need to go ahead and they need to be mature before the cell is actually going to go ahead and be ready for fertilization. So the first hormone that's generally released is the follicle stimulating hormone. The follicle stimulating hormone is going to go to the ovary. It's going to tell the cells, listen, you guys need to go ahead and you guys need to start maturing the egg cells. So one of the egg cells, from the great number of egg cells present, one of them is going to start to mature. Okay, as one of them is going to mature, they will eventually end up getting ready. Now, while the follicles are maturing, estrogen is going to be produced by the ovaries. Estrogen is going to do what? Estrogen is going to, meanwhile, it's going to be repairing and thickening and increasing the uterine lining. So estrogen is getting everything ready. Estrogen is slowly going to be thickening the lining of the uterus. Okay? And follicle stimulating hormone is going to be increasing the size of the follicle, right? It's going to be getting the egg cell ready for fertilization. Once the egg cell is ready, so what happens? That the concentration of estrogen increases. As more and more of the uterus lining will be laid down, the estrogen is going to increase. And as estrogen is going to increase, it will start to stop FSH. So estrogen, as it will increase, it will stop FSH. Why is it stopping FSH? Because FSH has done its job. It has gotten one of the eggs ready for fertilization. We don't want multiple eggs to be released. That does happen, by the way. That's how you get your twins. One of the ways you get your twins. But so estrogen will stop the effect of FSH, and as the concentration of estrogen will increase, so the lining will get thicker and thicker and thicker. Once the egg cell is ready for the for the for release, right? Once the egg cell is mature enough, so estrogen will stop FSH. It will say, FSH, you did your job. We have an egg cell perfectly mature. You need to stop working. And instead, it will go to the pituitary gland and say, okay, now we need LH. Now we need luteinizing hormone. So what does estrogen do? One, increases the thickness of the uterine lining. Two, tells FSH that you have done your job, you now need to stop. Three, it tells luteinizing hormone that now you need to start your work. Luteinizing hormone will come to the mature egg and it will cause ovulation to happen. The egg cell is going to be released from the ovary. Okay. Now, that's the function of luteinizing hormone, to cause ovulation. Once the egg has been released, it's going to be found within the body for about 1 to 2 days. Okay. So it's going to be found for 1 to 2 days normally. In fact, it's not even 1 to 2 days. It's around 24 hours mostly. So if within the small window, it's actually a quite small window, fertilization does not occur, right? So what is going to happen that if it does not occur, what is going to happen that your egg is going to be removed from the body. And after the egg has been removed from the body, obviously the uterine lining is going to start to break down. Now, what happens, thing that I want to add is, after ovulation, the cells of the ovary, we call them follicular cells. What do we call them? Follicular cells. Okay. These follicular cells become a different structure. This structure is known as corpus luteum. Okay. Corpus luteum. Corpus luteum is the structure. So this is basically the cells that are left behind after ovulation. The egg cell will be within the body for 1 or 2 days. In that process, progesterone that's being produced by the ovaries is going to continue to maintain the thickness of the lining. That's the function of progesterone, to make sure that the uterine lining stays intact, to make sure that it stays as thick as possible. So in that process, progesterone is going to maintain the uterine thickness. Then after within a day or so, if fertilization has not occurred, the egg cell will be removed by the body. Once the egg cell has been removed by the body, the corpus luteum is also going to break down. The amount of progesterone in the body is going to fall. And as a result, the body is going to go towards menstruation, in which the uterine lining is going to be broken down, right? In which the uterine lining is going to be broken down. And then the cycle will start all over again. If at this point fertilization does occur, what's going to happen? That the corpus luteum will take over the role of making progesterone. If fertilization occurs, corpus luteum will not break down. It will start to take over the role of it will go ahead and it will start to take over the role of making progesterone. So it will say, I will make the progesterone. I will make sure that the progesterone is present so that the uterine lining remains thick. Okay, since the uterine lining is thick, so now the zygote can implant itself into the uterine lining. And once the zygote has implanted itself into the uterine lining, eventually placenta develops. Once the placenta has developed, the placenta will start producing progesterone. Okay, so progesterone normally produced by the ovaries. What is the function? The function is the same, to maintain the thickness of the uterine lining. Then if fertilization occurs, the corpus luteum will take over the role for a few weeks, and then when placenta develops, placenta will take over the role of making progesterone. The function is to make sure that the uterine lining remains intact. So those are the four major hormones, and those are their roles that you're supposed to know about. Any question? Would anybody like to ask anything? All clear, guys? This is clear. I think, Maham, you wanted to go over it. I hope it's clear, no confusion. All right, so let's go ahead and we can take a look at the topic of our eye. So let's go ahead and take a look at that. Do let me know if anybody has any questions or if anybody wants me to go over something. All right, so going over the portion related to our eye, right? So in our eye, let's first of all review the different structures of the eye and what are the functions of these different structures. And then we have to go ahead and we have to talk about two reflexes, okay? So the structure of the eye looks something like this. In the front of the eye, we have a thin layer, a thin transparent layer known as the conjunctiva. The conjunctiva protects your layer, it keeps your eye, basically protects your eye and keep your eye safe from any dust particles, anything that can possibly irritate it. Then underneath the conjunctiva, we have our cornea. Our cornea is basically your bulging part of the eye, right? The part of the eye that is bulging out of your skull is basically where our cornea is. Okay, this is our cornea. Our cornea is basically responsible for refracting the light. It will refract the light, it will bend the light, and it will help in focusing the image. Then behind our cornea, you guys must have noticed that obviously your eyeball has a fluid inside it. This fluid is known as aqueous humor. Aqueous humor. And behind the aqueous humor, we have our lens. The thickness of the lens is very important. The lens can change its thickness, and that is the reason you guys are able to focus on objects that are far away and objects that are close to you. Okay? Then Other than that, we have two muscles. One muscle is the ciliary muscle, and the other one is the suspensory ligament. It's a muscle, but we call it suspensory ligament. The ciliary muscle and the suspensory ligament help in controlling the thickness of the lens. So they'll allow the lens to be thick or thin depending upon as needed. Then, other than that, so we have, this is the structure, the lens, the structure that is in front of the lens, right? The structure that is in front of the lens. This is the structure that is actually visible to us. The rest of the parts of the eye are hidden within our skull. Behind the lens, we have another fluid that we call vitreous humor. Then the entire, you can say, eyeball on the inside is lined up with your retina. Your retina is made up of rods and cones. Okay, it's made up of rods and cones. Rods and cones are needed for seeing different. For me to be able to visualize something, I need both rods and cones. When we talk about our rods, so rods are responsible for seeing color. If I want to see the color of an object, for that I am going to be needing my rods, right? Sorry, I think I said rods, I meant cones. Okay, the cones are basically needed for seeing your colored vision. Okay, so if a person is colorblind, you know where the problem can possibly be, right? The problem can lie within the cones. So cones allow you basically to adapt to the changes in light. As the light is going to be changing, the cones are going to be able to they are going to be able to adapt to the changes in light. Rods are the ones that detect the light. So the rods are the ones that are going to be able to tell that, oh, light is falling on the retina. Cones will be able to perceive color as well. Okay. Now, rods are generally located more towards the outside because I've seen questions on this. That's why I'm mentioning this, especially for the Cambridge students. I've seen questions on this. Rods are more towards the outside of the retina, and the cones are concentrated more towards the center of the retina. Okay. And when we go ahead and we talk about it, on our retina, we have a region known as the fovea. The fovea is the region where the image is actually formed. So the light rays are going to concentrate on the fovea, and this is where the image is going to form. Okay. So fovea has a high concentration of cones, by the way. Then we have a blind spot. The blind spot has no cones, it has no rods, and this is basically where the nerve, right? The optic nerve, the nerve that actually sends the information to the brain about what we're seeing. The optic nerve goes towards the brain. So these are the different parts of the eye. Now, other than the parts of the eye, there are two major topics in this thing that you guys should know about. You guys should know about the pupil reflex, and you guys should know about the accommodation reflex. What is the pupil reflex? If you guys stand in a dark room, your pupil is going to widen up. Your pupil is going to dilate. You step out into the light, your pupil is going to become narrow. It's going to constrict. That's the pupil reflex. I want you guys to remember the topic like this, that we are going to study about two muscles. We can have circular muscles and we can have radial muscles. Okay. Now, what is going to happen that whenever your pupil is dilating means that the size of the pupil is increasing, right? The size of the pupil is increasing. So what is going to happen that the circular pupil, the circular muscle is going to relax and the radial muscle is going to contract. As the radial muscle is going to contract, what is going to happen? This is relax, this is going to contract, and as a result, the pupil is going to dilate. The size of the pupil is going to increase. On the other hand, if you go into really bright light, the pupil is going to constrict. Now, the good thing about this topic is that the muscles are acting oppositely. So, if the circular muscles will relax when dilating, you know they will constrict when the pupil constricts. And if the radial muscles are contracting, they will relax. So, what happens is this, that the circular muscles relax and the radial muscles contract. This leads to the pupil becoming larger. So more light is able to enter the pupil, so you can see better. On the other hand, if you are in very bright light, the radial muscles will be the ones to relax and circular muscles will be the one to contract. The circular muscles are going to decrease the size of the pupil, so there's less light entering into the eye. Then we have the accommodation reflex. The accommodation reflex is the reflex that allows you to see things that are far away and things that are close to you. This is the reflex that is that becomes the problem that stops working properly, where people are not able to see things properly. If they can't see things that are far away or things that are close to them, it's a problem in accommodation reflex. Now, what happens in accommodation reflex? How does your eye see objects that are far away from you, and how does your eye see objects that are close to you? It does this by basically changing the thickness of the lens. Okay. You need two muscles to change the thickness of the lens. You need ciliary muscles and you need suspensory ligaments. Do not confuse them with circular muscles. Over here, I told you circular. Okay. For the pupil, we have circular muscles. For the accommodation reflex, for the lens, we have ciliary muscles. Okay. Now, what happens is this, that when I want to take a look at an object that is far away, okay, my ciliary muscles are going to relax. My suspensory ligaments are going to become taut. They're going to become tight, okay? They're ligaments. So they become tight, they become taut. And as a result, your lens is going to become thin. When your lens is going to be thin, so it's going to refract the light less. We are going to study refraction in physics, so it's going to refract the light less. Okay? Since it's going to refract the light less, so it's going to allow the light rays to focus properly on the retina and you can see the image clearly. If you are looking at an object that is close to you, now the light is coming from close to the eye, so it needs to be refracted more. It needs to be bent more because it's quite close to the eye. So what happens that this now the ciliary, so in this case, the ciliary muscles were relaxing. Now they will be the ones to contract and the suspensory ligaments will relax. The lens will become thick and it will refract the light rays more so that they can focus properly on the eye, and as a result, a clear image is made. So this is your accommodation reflex. So accommodation reflex allows you to focus on an object that is far away or an object that is close to you by using the thickness of your lens. Now, so this is what we had to know regarding our topic of eye. This is the major thing that you guys should know. You guys should know about exactly, how the accommodation reflex works, pupil reflex works, and what are the different parts of the eye. Okay. Any confusion, Ranim? I think you wanted to go over the question. I hope there is no confusion.
No, thank you, Miss. Understood.
Okay, great, perfect. Okay, okay. So, Arham, you wanted to go over your fermentation and yogurt making, so we can go ahead and we can take a look at that. Now, when we talk about our yogurt making, right? So yogurt making, I think this is specifically I don't think it's the part of the Cambridge syllabus, but it's there in the edX syllabus. So in your yogurt fermentation, what happens is this, that formation, what basically happens is that you are using the process of fermentation. Okay. How are you using fermentation? You are using fermentation to convert your milk into yogurt. Now, when we talk about fermentation, fermentation is basically due to an aerobic respiration, right? It's basically due to your anaerobic respiration. What happens in anaerobic respiration? In anaerobic respiration, respiration occurs, or I should actually say it better, in anaerobic respiration, glucose is broken down without the help of oxygen. This is done by your yeast. Yeast can do anaerobic respiration. That's what we see in bread making. And it is also gone ahead and it is done by your microorganisms. So your bacteria, a lot of your bacteria, a lot of your microorganisms are responsible for going ahead and going over fermentation, right? Now, how does the process of fermentation occur? Generally, we add a bacteria. The bacteria that we add is lactobacillus. Lactobacillus bacteria is added to the milk. Now, the lactobacillus bacteria is going to perform fermentation. How is it going to perform fermentation? It is going to convert the sugar in the milk to lactic acid. It's going to convert the sugar inside the milk, the glucose inside the milk, into lactic acid. Okay? Now, the lactic acid is what causes the milk to become a bit solidified, right? Solidified, and it causes it to also become sour. That's the reason why it has a sour taste, due to the formation of your lactic acid, right? Vinegar also tends to have a sour taste. So this is the reason why you end up seeing that it ends up having a sour taste because of the production of lactic acid. So for the formation of yogurt, what we generally do is that you generally start with pasteurization. We do that. We do pasteurization, kill any harmful microorganisms. Then we add lactobacillus. You heat it to around 40 degrees centigrade, and then you are going to go ahead and use your you're going to heat it to 40 degrees centigrade, and then you're going to go ahead and the bacteria is going to convert the lactic acid, the sugar into lactic acid. This lactic acid will then solidify it, giving it a curd-like structure, and also go ahead and give it the sour taste. So that is how we actually form our yogurts. Fermentation is also, by the way, used for raising, making bread and all, right? Fermentation that is done by yeast cells is different than the fermentation done by this bacteria. The bacteria converts sugar into lactic acid. Yeast cells will convert your sugar into ethanol and carbon dioxide. So you can see different types of fermentation depending upon depending upon what organism are you using. If you use yeast, you are going to see that the lactic acid will be produced. And if you're using if you're using lactobacillus, you'll see lactic acid is produced. If you use yeast, you see ethanol and carbon dioxide is produced, right? So fermentation is, quite simply put, the breakdown of glucose without the help of oxygen. So it's done in anaerobic respiration. Aram, got it? Clear? Any confusion?
I know, miss. Thank you.
Okay, all right. Okay, now I was thinking, let's go ahead and just go over some exam questions and go over how we're actually going to solve some questions related to the topic. So let me share the questions questions with you. Let's go over some questions and see what kind of exam questions do you encounter. What exam questions do you actually see in your papers, and how are we going to solve them? How are we going to do them? Is there any particular topic that you guys feel is the questions tend to be harder? Like you guys know the topic, but the questions are confusing. Let me know
Miss,
in the chat.
Miss, the
um,
answer is
Kiara?
wrong.
experimental questions, like where they ask you to
Okay, Arham.
describe a certain experiment. I think poems.
Mm-hmm. Okay, all right. Anything else?
Miss, the Chrome experiment, the one like you have to like describe an experiment.
Right. Okay. Okay, all right. I'm going to try my level best to see if I can find some questions like this and I can go over how to answer them with you guys. Okay, now this is a very nice question, by the way, that is related to the topic that we just discussed today. So let's go ahead and go over it and see if we can answer questions related to it, right? I want you guys to go ahead and tell me what do you guys think the answer should be, okay? Because we discussed the topic together as well. So we have figure 2.1 is a diagram of a part of a DNA molecule. They're saying draw a circle on figure 2.1 to identify one pair of bases. Would somebody like to annotate on the screen? Can you identify a pair? Please keep in mind the question says pair of bases. Can somebody let me know? Can somebody annotate what a pair of a base should be?
Yes, maybe.
Yes, yes, correct. That is a pair. Now, the next question is interesting. I don't know if you guys have gone over questions like these. They're saying the percentage of T bases in the DNA of a species is 29%. Calculate the percentage of bases that would be the base G in DNA of this species. These questions are easy to solve, but there's just a small trick to them, right? Let me go ahead and tell you how to solve questions like these. We have adenine, thymine, cytosine, guanine. 29% of the DNA is thymine. Now, remember, adenine and thymine are complementary to each other, which means that the amount of thymine has to be equal to the amount of adenine, right? Because they're complementary to each other. They only make bonds with each other. They only pair up with each other. So they have to be equal to each other. If one is less than the other, then how is the DNA going to form, right? Similarly, let's say that we don't know how much guanine is in the body. I want to find out. That's my question. That's what they're saying. Find out the value of guanine. So if guanine is X, that means cytosine is also X. Because again, guanine and cytosine are the same, are going to be equal to each other, right? Because they pair up with each other. So they're going to be equal to each other. So cytosine is X, guanine is also X. Now, we know that the total has to be 100%. So we will go ahead and we will solve for X. Adenine plus thymine plus cytosine plus guanine is equals to 100%. So adenine is 29, thymine is also 29. Cytosine, we don't know how much cytosine we have, it's X. How much guanine do we have? X. So 29 plus 29 plus 2x is equal to 100. So can somebody please solve for x and let me know how much guanine is going to be found within the sample?
So miss, we add 29 with 29, and then we get the answer, and then we divide it by 2.
First subtract from 100 and then divide by 2, right? So you'll do 29 plus 29. That's going to go ahead and that's going to give you,
58.
yes, so 58 plus 2x is equal to 100.
And
So
then
move this to
that's
the other side. Yes.
42. And then we divide
Yes.
42 by 2, which is
Yes.
21.
Yes, exactly. So that means that how much guanine do we have? We have 21%. Okay. So this is how you are going to go ahead and solve questions where they ask you to find out the percentage of bases. Okay, now they're saying state the name given to a length of DNA that codes for a protein. You guys know the answer to this, I know, but you guys might still feel a bit confused. What should I call a length of DNA? What the length of DNA that codes for a protein? The length of DNA that codes for any protein. Yes, gene, correct. That's what gene is. Gene is the length of a DNA that codes for a protein. Now,
Miss, can
next
you go up?
question.
I'm gonna copy the question.
Yes, definitely. Do you want me to go up over here? There's nothing over here as such though.
No, just the down part.
Yeah, here you go.
Thank you.
Okay. Now, the next part is what we discussed as well, and this is a 5-mark question. Okay. Explain how proteins are made by the cell. Take a minute. I want you guys to take a minute, think about it, and then let's see what answers would you like guys like to give. How do we make proteins? How does a cell make protein? Yes, okay, so I put a good answer over here as well. Would anybody like to, so keep sending your answers. I'll discuss this with you guys in a second. I'm just waiting for everyone to send me the answers. Okay, I've got another answer over here. Good. Okay, another answer. Okay, so I've got some really good answers over here. Okay, I've got another one. Okay, all really good answers. So now what are the steps that I want you guys to go ahead and you can mention in the exam? Let's discuss. I've got some pretty good answers from you guys, by the way. That's a very good job. Well done. Now, you're going to start with first talking about obviously transcription, right? DNA has the base sequence that codes for the protein. So we need a way of coding the base sequence. So what are we going to go ahead and do for that? For that, we're going to do transcription. So and you can add this point. Since DNA is too large to leave the nucleus, the DNA is transcribed and a messenger RNA is made. The messenger RNA has the triplet code, right? The messenger RNA will move out into the cytoplasm where it will bind to the ribosome. The transfer RNA with the complementary anticodon will bind to the triplet code and bring the specific amino acid with them. The amino acid will join together to make a protein. So these are all of the points that you guys can go ahead and include in your answer. DNA is too large to leave the nucleus, so it is transcribed. Messenger RNA is made. Messenger RNA has the triplet code. So the transfer RNA has the anticodon, right? It has the complementary matching anticodon. So the transfer RNA who has that codon, only that transfer RNA will come and bind to the messenger RNA. And it will bring the specific amino acid. Whatever amino acid was needed, it will bring it accordingly. The amino acid will then join together with each other and a protein will be made. So really good attempt by you guys. By the way, that was a very good job. I really like the answers that you guys sent. Okay, let's take a look at another interesting question.
Miss, was that a 6-mark questions?
It was for 5 marks.
Okay.
Okay, so Shama wants to know, how do we write down the process of carbon or nitrogen cycle for 6 marks? Okay, let's go ahead. We can take a look at that. How do you answer your nitrogen cycle question as well? Now, when we are going to go ahead and talk about your nitrogen cycle, right? So the nitrogen cycle, especially I'm talking about the nitrogen cycle because nitrogen cycle is has a lot of different parts related to it, right? We have nitrogen fixation and everything like that. So let's see. Whenever you guys are writing down the answer for nitrogen cycle, these are the points I want you guys to make sure that you include. Talk about nitrogen fixation, right? You are going to talk about nitrification. you are going to talk about assimilation. You are going to talk about your denitrification. So all of these points you guys should make sure are included in your answer. Not only should these points be included in the answer, but always include a 1 to 2 mark definition of exactly what this is. So this first, for example, we can say it's a cycle. So technically in cycles, we don't have first steps, but the first step for our discussion is let's talk about nitrogen fixation. Okay. What is nitrogen fixation? So in the process of nitrogen fixation, the nitrogen gas is basically converted just give me 1 second. Sorry, guys. So nitrogen gas is going to be converted. Now, what basically happens is this, that with our nitrogen, there's a problem. Nitrogen is pretty non-reactive, right? Nitrogen is like really non-reactive. It does not react easily at all. So my plants want nitrogen. How do they get the nitrogen? For that, they somehow need to take that inert nitrogen and convert it into something that is more usable, right? So for that, what is going to happen that we have got certain bacteria that are found within the soil. Those bacteria are going to go ahead. They are going to convert your, they're going to convert your nitrogen into ammonia. Okay, that's nitrogen fixation. So you'll start by first talking about nitrogen fixation, that bacteria in the soil is going to be converted, is going to convert the nitrogen that is found within the air into ammonia. Okay, now the thing is that ammonia is easy for the plants to use. The plants can still use ammonia for their growth, right? Now, the next step that happens is nitrification. In nitrification, what are you going to go ahead and do? You are going to take that ammonia and convert it into nitrate, right? We need nitrate, potassium nitrate, sodium nitrate. We require nitrates. So the ammonia is converted to nitrates by bacteria through oxidation. So the bacteria perform an oxidation reaction, which you might have studied in chemistry. And the ammonia is going to be converted into nitrates, right? Then after that, after the ammonia has been converted into nitrates, what is going to happen? That these nitrates are going to be absorbed. Nitrates are absorbed from the soil by the plants. This process is known as assimilation. The plants are absorbing the nitrates from the plants, right? Now, once the nitrates have been absorbed by the plants are going to grow. These plants are going to be eaten. The plants are consumed by animals, right? They are consumed by animals as well. So the nitrates pass to the animals as well. The animals will also end up getting nitrates as they're going to eat your plants. And then what is going to happen that once the plants are going to die, once the plants die, once the animals die, right? Or even through excretion, even though excretion from the animals, right? What happens that this nitrate is returned back to the earth, right? The nitrates ions are returned back to the earth, right? So this process is known as ammonification. So the nitrates are released back into soil as due by the decomposers. So your decomposing bacteria, decomposing microorganisms are going to break all of this down, and the nitrates are going to be released back into the soil, right? The nitrates will be converted back to ammonium salt, your potassium salt, put your ammonium salt, your NH4 plus, right? NH4 plus is your ammonium. It's going to be converted back into that. And finally, we have denitrification. So in denitrification, you are going to go ahead and convert the ammonia back into your nitrogen gas.
Miss, denitrification is done by bacteria, right?
Yes, all of this is done by
Okay.
bacteria.
Okay.
So denitrification is going to convert the nitrogen back into your nitrate ions, your ammonia back into nitrogen, and the nitrogen is now returned back into the air. So the cycle can start all over again. So this is an answer that you can go ahead and you can give for your nitrogen fixation. Okay. Now, let's see. There's another question that I just want to go over with you guys. Please give me 1 second. Let me see if we can find any questions on planning experiments. You guys wanted to do that? If I find one, I'll definitely go ahead with you guys. Okay, take a look at this. So our question says urea is a waste product. Describe how urea is formed. This is very interesting. How is urea formed? I don't think I mentioned to you guys when I was teaching excretion how
It's
urea is.
excess of amino acid
Yes,
from the liver.
yes, it has everything to do with amino acid. Yes.
Miss,
Yeah,
wait,
it's a
Miss, how
3-mark
it have
question.
amino acids? Miss,
Sorry?
isn't urea like
Urea is
how is ammonia there in urea?
how is amino acids, not ammonia, amino acids. Your
Okay.
amino acids, because amino acids contain nitrogen inside them, right? Urea also contains nitrogen inside them. So the liver is the one that is actually responsible for making urea. The liver, it actually goes ahead, it takes those amino acids and it breaks them down, right? Because the body, whatever amino acids the body wants to use, it will use up. But what about the amino acids that are excess? What about the amino acids that the body needs to get rid of? So what is the body going to go ahead and do in that case? So in that case, the body is going to go ahead and it's going to use your, it's going to go ahead and it's going to use liver. So the liver actually breaks down your amino acids. So when amino acids are broken down, that's how you basically end up getting your urea from. That's where the urea actually occurs. So liver is actually quite important for your excretion. Okay. So the liver will break down any excess amino acids and it's going to produce urea. Okay. And not only that, the liver is also responsible for breaking down hemoglobin, by the way, right? But okay, in the topic of circulatory system, you guys must have studied that in the circulatory system, red blood cells do not have a nucleus, right? Why do they not have a nucleus? To allow for as much space as possible for the for your hemoglobin to be carried. Now, because they do not have a nucleus, so they are obviously have a very limited lifespan. Red blood cells have a lifespan of about 120 days, which is around 4 months. Then after 4 months, they're broken down by the body and body makes new red blood cells. So in the process, hemoglobin is broken down. The hemoglobin that is broken down by the liver as well. So the liver is the one that's going to be responsible for breaking down hemoglobin, and it's also going to be responsible for breaking down your um, amino acids as well. So the amino acids are going to be broken down, and that's how urea is going to be made. So that is the that's basically the answer to the question that I was looking
Miss,
for.
can you write it down?
Yeah, I will definitely go ahead and write it down. Urea is formed by the liver in a process known as deamination. Deamination, that's what the process is called. In this process, the nitrogen containing part of the amino acids is removed and used to form urea. That's what you can write down for 3 marks, right? So the nitrogen containing portion of the amino acids is removed, right? And is used to form urea. Hence,
Miss, do I need to write the process name in Excel?
you don't need to write it down, but if you mention it, that will be one thing in your favor. Hence, excess amino acids are removed by the body. Okay, now next question. State the component of the blood that transports urea. Does anybody know what part of the blood transports urea?
Plasma.
We have different, yes, plasma. State why urea must be excreted. Why do we need to get rid of it? Because it's toxic to the body. We
Toxic?
cannot, yes, it's toxic to the body. We cannot keep it inside the body for too long. It's harmful for the body. It's toxic. We need to get rid of it. Okay, state the name of the blood vessel that carries the blood away from the kidney.
Vein?
Yes, vein, but specifically renal vein, right? Kidney is renal. The term we use for kidney is renal. So renal vein. Renal vein carries the blood away from the kidney. All right. Now, figure 4.1 shows the drawing of a nephron in the human kidney and the associated blood vessels. The question to us says, describe how the structures labeled in figure 4.1 produce urine. So this is for 4 marks, right? You have to discuss exactly how these structures are responsible for producing your urine, right? Okay, so first of all, can somebody tell me what the labeling is?
Miss K is glomerulus.
K is your glomerulus, correct. So this is the Bowman's capsule. This is the Bowman's capsule. This, okay. And the capillaries that are present inside the Bowman's capsule, we call them the glomerulus. Good job, Aram. So we call them glomerulus. The capillaries inside the Bowman capsule, we call them glomerulus. So K is actually your glomerulus. What is N?
Collecting that?
Collecting dark. Good job. What is M? And pay attention to exactly where the arrow is pointing.
Is it a blood vessel?
It is the blood vessel. This is not pointing to the loop of Henle. It's pointing to the capillaries that surround the loop of Henle. So this is your capillary. Perfect. So,
This one
and
takes
what will
blood away from the body or to the body.
It's basically there for selective resorption. So it will take away the nutrients, whatever nutrients need to be absorbed back to the body, and then it will return it back into circulation. So one part of it is like when you actually look at the diagram, half of it is capillary and half of it is vein. The main function is to basically to make sure that whatever substances need to be absorbed back, they will all be absorbed back, and then it will return it back to the body. So all of the blood from here is going to
Yes.
go back towards the heart.
Can you
Yeah.
put the PDF on WhatsApp?
Sorry, can you say it again?
Can you put the PDF on WhatsApp?
Sure, I will. Okay, so your question, and what should I call L? Can anybody tell me what L is?
PCT.
No, it's the nephron. That's it. You don't have to worry about the further labeling of it. You guys don't need to worry about any further labeling.
Okay.
Just say this is a nephron. That's it. Now, so now go ahead and take 2 minutes. Go ahead and list down exactly how these structures will now be responsible for making urine. And we will just discuss the
Yes, can you go back to the question?
Yes, absolutely. If you guys need to see the diagram, let me know.
Miss, could you go to the diagram?
Yes, here you go. All right, so I've got my answers. Let me go through them. Right. Good answer. Arham, good answer. Okay. Now let's go ahead and discuss let's go ahead and write down the answer so everyone can go through it. So when we're going to go ahead and we're going to talk about it, this is how you can go ahead and you can answer the question, right? The glomerulus, K, that's our K, are the bunch of capillaries that are found in the Bowman's capsule. The capillaries help in ultrafiltration, where, right, they help in ultrafiltration. Okay, water, glucose, urea, water, glucose, urea, hormones, toxic substances move into the nephron. And as they move into the nephron, what is going to happen? The nephron, that's your L, will perform selective resorption. In this process, glucose, your glucose, some of the water, your mineral ions will be absorbed back into the bloodstream, right? The urine will concentrate and eventually pass to the collecting duct. So this is how you guys can go ahead and answer the question. So so eventually urea, excess mineral ions, excess water, hormones and toxic substances are removed. So uh this is down. I'm sorry, this is not found, this is down. So this is how the process is going to happen, and this is how you're going to go ahead and answer some of the questions. All right, so guys, we'll go ahead and we will end it over here for today. Once again, I hope you guys enjoyed this session. I hope that it was helpful for you. I really hope, inshallah, I'm actually praying for all of my students that inshallah you guys do a wonderful job. Best of luck for exams. I know you guys are going to have it soon. So I hope inshallah it goes really well, and I hope this really helps you guys out and you guys found this session to be useful. Once again, just a reminder, we are starting our next batches as well for your October, November, as well as for your next year, 2024, May, June. We are starting our batches very soon. We are starting the batches for your May, June, in August. Okay. We have very limited seats. We only take 3 students per batch. So please go ahead and join up. I hope I will see you guys again for my A-level classes as well. Best of luck for your exams, guys. I hope inshallah you all do really, really good and you guys enjoyed this session. Thank you so much, everyone. Have a very good day.
Thank you, Miss.
Bye,
Thank
guys.
you,
Thank
Miss.
Okay, miss. Thank you. Allah Hafiz.